So what we're going to do, we're going to get back to where we started. When we talked last Thursday, we specifically talked about blood. And like I said, that's fairly straightforward. When we get into how hemoglobin works, that is what we're going to be talking about in our vessel lecture. So right now, we're just honing our focus on the heart. So by now, you guys saw this slide. This slide covers the pathways. Again, stuff that you would have covered in anatomy. And we're looking specifically at the pulmonary circuit, systemic circuit, which is okay because we're going to revisit this again in our vessel lecture when we start that at the end of Thursday. If we have time, I believe we should be able to start this on Thursday. We'll talk about the different vessels. Where we want to really hone in our focus on is the heart. So today, we are going to be covering the cells. So we're going to start back at our cellular layer, and we're going to first take a look at how these two types of cells that are prominent in the heart are going to work electrically. So we're going to have to go over all that stuff when we looked at depolarizations, repolarizations, in order to understand the electrical excitability of the cells, and then what the impact is on the cardiac muscle cell. So what we're looking at are the pacemaker cells. These are specialized cells that are attuned to being autorhythmic. And what do we mean by autorhythmic? They, if you were to take a pacemaker cell, put it in a petri dish, give it enough oxygen, give it enough nutrients, those cells will beat, and they'll beat on their own. Okay? And their whole purpose is to just beat. They are the sort of the conductor of the heart. They tell the heart when to fire, and they do it because that's exactly what they do. They're pacemakers of the cells. Now, they don't do anything else but that. They don't contract. They are limited in terms of their contractile property. In fact, if you were to take a look at the image on the bottom, you can see there's some presence of striations, which gives you shows you the ability that these cells can contract if they need to, but those things have been inactivated. Really, their entire purpose is to lead the contraction so that the heart can pump in a uniform fashion. And we'll talk about that as we start to talk more about the electrical excitability. You'll learn more about how the cardiac muscle cells will contract, and then we'll take that overall and take a look at how the heart tissue will beat in this fashion in order to get blood to pump efficiently throughout the heart. So that's the pacemaker cells. The other portion of the cardiac muscle cells, where we're going to again look at the action potentials that are generated in order to result in that muscle contraction that takes place. So we'll take a look at the properties of that fairly soon. So cardiac muscle cells, if we're to take a look at them, again, we didn't spend too much time on our muscles, but the main characteristic of our cardiac muscle cells is that they're branched. You can see the branching that takes place, a lot of different wide branches. They're short, and they're fairly wide cardiac muscle cells. You'll see in the book, they do refer to different names: myocardiocytes, myocardial cells, myocytes of the heart, or simply put, cardiac muscle cells. So the whole, by looking at all of these terms, you can see they all have something in common: myo, cardio, and then cytes, meaning cells. So whether I call it myocardiocytes, I call it cardiac muscle cells, myocardial cells, you get the general idea. These are going to be the contractile cells. Pacemaker cells are going to be called pacemaker cells. So you know exactly what I'm referring to. These are the non-contractile, these are the contractile cells. Looking at the cardiac muscle cells, you can see that on our images to the right, these junctions in between adjacent muscle cells are connected together. And you can see that on our cells over here. You can see these, what are called intercalated discs. Basically, these cells are zippered together. In fact, the way they're zippered together, they have desmosomes. And if you remember from our tissue lecture, desmosomes serve for the purposes of anchoring the cells together. That keeps them stuck together. Now imagine if these cells didn't have anything that anchored them together, these cells are constantly contracting. And over time, they could start to break apart. So we want something that glues them together tightly, and that's exactly what we want. So in between, in these intercalated discs, what you'll also see, aside from the desmosomes, you're going to see these gap junctions. And if you remember, again, from our tissue lecture, the whole purpose of those gap junctions are to allow ions to move through. So what we've been learning about neurons, when neurons communicate with one another, you'll see the electrical activity that takes place within this neuron number 1. You see a neurotransmitter diffuse across, that's the chemical portion, and then it makes its way over, that neurotransmitter targets the next neuron, binds to a receptor, causes electrical activity to take place on the second neuron. Here we get direct ions moving from one cell to the next. So if we were to see a wave of depolarization, that is a rush of sodium starting in this one muscle cell, those sodium that make their way out from the extracellular fluid into the intracellular fluid will start to rush through those gap junctions. And that will cause excitation on those adjacent muscle cells. So you'll see how those waves take place, and that's how we can have electrical activity take place very quickly. There's no diffusion across any synapse. It's direct ion transfer, so direct electrical transfer from one cell to the next. Now, the other thing to take a look at the cardiac muscle cells is that we have high mitochondrial density. And by that, we know exactly what the mitochondria is responsible for. We know it's for the generation of ATP. We know it's going to undergo glycolysis. It's going to use those mitochondria in order to generate that ATP efficiently. So therefore, it's not really using anaerobic. It's not primarily dependent on anaerobic metabolism, i.e. glycolysis. It needs to undergo the Krebs cycle, undergo the electron transport chain in order to efficiently make energy. The other thing too is what, and this is covered in the book, but not really important for the purpose of the lecture. The heart doesn't only use glucose, it can also use lactic acid as well as a byproduct. Therefore, it does efficiently, obviously it's a critical tissue. It needs to work, and it needs to generate ATP to continually pump blood through. So it's going to use any metabolic substrates it can in order to generate optimal amounts of ATP. Now, I did mention earlier, on the earlier slide, you saw prolonged depolarization. That's the main characteristic of our muscle cells that you'll be able to see when we get into our membrane potential, the changes in the membrane potential on our later slide. I know in all of our neurophysiology lecture, when we talked about our neurons, we talked about sodium and we talked about potassium and the movement of those ions. The third thing we're going to see that's characteristic in our cardiac muscle cells are the calcium channels. So we're going to see how these voltage-gated calcium channels are going to be involved in prolonging that depolarization. Looking at the cardiac muscle fiber arrangement, here's just an interesting slide behind it, just graphically showing you how all the muscle fibers, you're going to see our actin and myosin, which you learned in anatomy, are all creating these cross bridges to basically allow the contractile tissue, the cardiac muscle cells, to contract one another. So here you can see the different fibers, the different fibrous proteins that create bridges together. And I'll be able to show you as we go through the generation of tension that takes place. At the start of depolarization is going to start to create these bridges that are pulling one another and effectively sliding. So here you see the sliding filament theory in order to shorten the muscle fibers that you see on our prior slide, in order to shorten these but to also elicit contraction. So keep that in mind when we start looking at not just that individual muscle cell, but how all these cells operate together to create contraction that takes place in that tissue. Now here we have an exercise. So what I want to point out in this exercise is that while we have here these hexagons are a bunch of muscle cells, cardiac muscle cells. And in between here, you'll see the gap junctions. Each of these represent gap junctions. What we have are just and you can just think of just a blanket sheet of a bunch of cardiac muscle cells that are all arranged together, and they're all held together by their intercalated discs with gap junctions that allow the transfer of ions in and out. And so take these muscle cells by themselves, and like I said earlier, put them in a dish, give them enough oxygen, give them enough glucose, and they'll beat on their own. And they'll continually beat as long as you provide enough nutrients for them. Now, each of these by themselves, these cardiac muscle cells, beat about 20 to 30 beats per minute, and you'll be able to see that when we talk about that in a later slide. If left on their own, 20 to 30 beats per minute, and it's not like individual cells are beating at their own rhythm. What's going to take place is you're going to take one cell And one cell that starts to depolarize, you remember in our action potential, the first phase of the action potential is what? You guys tell me. What ion is rushing into the cell? Sodium, right? So we're going to get sodium, extracellular sodium. And I'm just going to start, I'm going to pick on this, this cell right here. And we're going to get sodium rushing in. That's going to be the first phase of the action potential. And that sodium that rushes in is going to start moving through those gap junctions. So while this cell depolarizes and undergoes that first wave, those sodium ions are going to start moving over, and this is also going to depolarize nearby adjacent muscle cells. So as this starts the wave, then more sodium comes in here, more sodium comes in here, this depolarizes, brings this to threshold. And as this starts to reach threshold and causes more sodium to rush in, all the other ones coming surrounding will also start to depolarize. So what starts off as this cell firing creates waves that propagate away from that single point. Now take this muscle cell, strip it of all of its contractile proteins. The whole sole purpose of this is now that if this beats at 20, 30 beats per minute, all of these will beat at 20, 30 beats per minute. So this is going to start that point at which contractions, at least when our depolarizations will take place. So this cell, we want to reprogram. We want to remove the contractile proteins, and we're going to tell this cell to beat at about 100 times per minute. That means even though the other cells can beat at 20, 30 beats per minute, every time this fires, everybody else fires. So this reprogram is exactly what happens with our what's called our pacemaker cells. These pacemaker cells are geared to contract and be the pacemaker cells. They're the ones that lead when the rest of the nearby cells will fire. So looking at these pacemaker cells, like I said, they're non-contractile. They have the ability to spontaneously depolarize. So they beat at 100 beats per minute, and they tell when everybody else beats.
So those are pacemaker cells. Earlier you said they were cardiac.
Okay, so they all of these are cardiac muscle cells. I'm just pinpointing this one and saying this is the one that's going to be the one that's most excitable. If you reprogram it, and that's basically what's happening. When you look at cardiac stem cells, they can either be pacemaker cells or they can be muscle cells. And these cells are going to be the one that's reprogrammed to be only the beating cell. Okay, everybody else is going to contract. They do the same thing. You're going to see the same action potentials, but there's going to be a slight difference in those action potentials, and that's what I'm going to go into in a little bit. So these cells are specifically the pacemaker cells. And if you were to wrap this layer over a heart, you can see that there's going to be a specific area that will beat at 100 beats per minute, a specific group of cells called the SA node. And that SA node, which we'll discuss later, is called the sinoatrial node. And as you can surmise from the name, it's going to be located towards the atria, and I'll show you that on our next slide. So these SA node cells are pacemaker cells. They don't contract. All they do is they spontaneously depolarize. This is the term which we refer to as autorhythmicity, meaning automatic rhythm. And as these fire, this is going to create the rush of sodium ions that move all the way through. Every time this beats, sodium rushes in. Sodium is going to move through those gap junctions and travel down all the branched cardiac muscle cells that they innervate. So let's take a look on the next slide over. What we're going to do is we're going to talk about what that membrane potential looks like. So here we're going to first look at, these are going to be our pacemaker cells. And I've got this all drawn out for you on the next couple slides, so don't worry about having to draw for accuracy. I'm going to be referring to each of these steps below. So if you want to just kind of listen through, we can start from there. Now you'll see that most of the cells we're going to start off with, you'll see our threshold that's drawn in blue. We're going to start off at about negative 60 millivolts as our resting membrane potential. These are our pacemaker cells that are at rest. Same principles apply as to why it's negative 60, negative 70. Again, large anions inside the cell. You've got our sodium-potassium pumps, and you have leaky potassium channels that also contribute to why it's negative 60 as resting membrane potential. And what we're going to start off with, again, is at our starting point right at 0 seconds. And we're going to watch how this pacemaker responds over time. Now, the critical thing that you're going to be able to see about our sodium channels, and you can see this on our first slide over, the very beginning is the presence of leaky sodium channels. Now, think about what happens. And I know I mentioned leaky potassium channels. Those are still there, but we have more prominent leaky sodium channels. So what does that mean? We've got a cell on the outside. We've got leaky sodium channels on the cell membrane itself. That means when our cell is at rest, we've got a high concentration gradient of sodium on the outside wanting to be driven through the inside. So this means as the cell is at rest, we're going to see sodium going in. And as sodium goes in, we know that that's going to be decreasing the membrane potential. And when I mean decrease, it means bring it up towards 0. So there's going to be an upswing. And so it's going to just slowly, based on the number of sodium channels open, will allow this continual rise in the membrane potential, up to the point that it reaches negative 50. So by just virtue of the number of sodium channels open, this will continually rise and rise and rise. But right at the point of negative 50 is we're going to notice this point, we're going to see a huge onslaught of a depolarization curve. So what started off as this initial portion is sodium entering through the leaky channels. This second portion, this next upswing, is going to be calcium channels. So calcium channels opening at that point. Our voltage-gated calcium channels will now open up, and that's going to allow now calcium, which is high extracellularly compared to that intracellularly. We have calcium rushing in, and that's going to further depolarize. So now we see the added effect of calcium, a positive ion. We see positive sodium ions coming in. We see now a huge rush of calcium, positive ions coming in as well, because we've opened up those voltage-gated channels at threshold. Once we get to the very top, and we're going to get closer to about 0 to about 10 millivolts, right up at that very top, you're going to see calcium channels close. And then we're going to start to see potassium channels open up. And knowing that potassium is leaving the cell, again, high concentration of gradient on the inside, wanting to leave the cell, our voltage-gated potassium channels open, and then we're going to repolarize. And that's going to keep repolarizing. We're going to get to the point where we repolarize close to resting membrane potential, and then we start back at where we started. Right at this point, our potassium channels close, and we start again. Again, we're back at resting membrane potential. Our leaky channels are always open, and then it takes us back to threshold. At threshold, calcium channels open, rush calcium in, potassium out, back to resting membrane potential. This slope right here that I'm referring to is what we call our pacemaker potential. And the rate of that pacemaker potential, how fast do we start from resting membrane potential all the way to threshold, is dependent on the number of sodium channels open. So we'll look at that. If I were to open up more channels, you would notice that the rate, and let me draw this in blue. If I increase the rate at which our pacemaker potential reaches threshold, you'll see that the action potential will take place even faster. And each time we have an action potential take place is a heartbeat. Like I said, it's a pacemaker that's going to start that wave of depolarizations for those adjacent cells. So in that portion where you saw 1 in red, our pacemaker's lower rate of change in our pacemaker potential, then we see 1 action potential take place. Here you're able to see 2 because we see an increased rate. So this difference, everything below threshold, our pacemaker potential is what's going to determine when that action potential takes place, and therefore that wave of depolarization for the rest of the muscle cells. What questions do we have about this?
So what is pacemaker potential is just basically
The rate at which we reach the threshold. It's,
because you're opening up what channel?
yeah, you're opening up more sodium channels.
Also,
Yeah. So the more sodium channels that are opened, that means that's the rate at which we reach threshold. So more sodium going in brings us more and more and more towards depolarization, and eventually we reach threshold, we open up those calcium channels.
So it doesn't matter if we increase the calcium channels too?
Because once we open up those voltage-gated calcium channels, then you're going to undergo that whole rhythm. You're going to undergo the depolarization to repolarization. So this is where calcium comes into play. What you learned last time was the action potential where sodium rushes in and potassium comes out. Calcium just comes in to add to the pacemaker potential to really cause that deflection. This is going to be the huge wave at which once this takes place, once we get this action potential that takes place, that constitutes 1 heartbeat. Then we're going to go back, and then the rate at which we reach threshold again is going to see the number of waveforms that take place. That answer your questions? So looking at all of these will basically show you, again, everything that I've drawn. Potassium channels closing, slow sodium channels opening, sodium entering, calcium, potassium, again. All of that leads to our action potential. And again, the next slide over breaks that down for you. So in reality, we don't really see a resting membrane potential that's stable. And that's all due to those leaky sodium channels. Anytime you have a leaky channel open, it's just going to allow things to move down its gradient. There's nothing regulating those ions or preventing those ions from coming through. And for calcium, however, on the other hand, those voltage-gated channels, we really need the right voltage in order to trigger those the gates to open, to trigger an action potential to take place. So like I said, in that image earlier where I had drawn out all the heart cells, the area that's going to be the most sensitive, that's going to lead the pacemaker cells, are going to be the sinoatrial node. And the sinoatrial node is actually a very small area. It's about, I believe, like 2 to 3 millimeters of an area of concentrated pacemaker cells. And like I said, they beat on their own. Give them enough nutrients, and they will beat. And dependent on that pacemaker potential is the rate at which those sinoatrial node, which contains those pacemaker cells, will depolarize. And this, if you were to count the number of beats, or how long the beat takes, you'll see this is about 0.8 seconds. So 60 seconds divided by 0.8, that's not exactly 100. We'll just pretend it's about 100. It's about 100 beats per minute. What we'll see on here, you'll see no CNS innervation, no central nervous system innervation necessary for depolarization, which is true, that if you were to take a heart, and I don't know if you guys ever played like Mortal Kombat from way back in the days. They had that one character, I can't remember who, but they would like reach in, grab the heart. That heart was still beating. Actually, a heart would still technically do that, because it doesn't need the central nervous system for it to beat on its own. Those sinoatrial node contains those pacemaker cells that will beat and still cause the heart to beat on its own. Eventually, it's going to run out of nutrients, and it's going to run out of O2 delivery to that area. Those cells will fail, and then the rest of the other cells will fail as well. So it won't continue indefinitely if that's where it needs blood coming in from the coronary arteries coming in and feeding the rest of the heart. And as I said, the SA node doesn't contract. It fires on its own, but it doesn't have any contractile properties. Now, the sinoatrial node, the SA node, isn't the only place that contains pacemaker cells. In fact, the entire conduction system of the heart, which I'll describe in a little bit, also contains pacemaker cells. So I mentioned earlier, this is the SA node, but everything else that you see in red right here, there's a whole highway, a conduction highway, that travels from the atrium down the interventricular septum, which is going to be that muscle mass in between that separates the right from the left ventricles. And then it's going to wrap down to the apex of the heart and travel back up, called the Purkinje fibers. And that's all described for you on the next slide. The conduction system, again, consists of all pacemaker cells that can also fire should any of the prior nodes stop firing. So let me pause right there and let me first describe the entire highway on its own. So sinoatrial node fires about 100 beats per minute. That means every 0.6 seconds that the SA node will fire. And that wave of depolarization travels to all the adjacent muscle cells nearby. And eventually, through those gap junctions, you're going to reach the next node over. And that node is about 0.3 millimeters away. Millimeters away from one another. The next node is called the AV node, which is called the atrioventricular node, and it's located really at the most posterior portion of the right atrium. This node right here, like I said, about 0.3 millimeters, very small delay, creates this delay that travels from the SA node over to the AV node. Once that depolarization wave makes its way down to the AV node, then it's going to move, it's going to travel all the way down what's called the bundle of His. And again, that separates the right ventricle from the left ventricle. That bundle of His will then move down the left and right bundle branches where it'll separate. You've got the left bundle branch, and that's going to travel down the interventricular septum, down to the Purkinje fibers. Purkinje fibers are the ones that wrap around, and then it makes its way to the outer, the lateral sides of the heart. So looking at this wave of conduction, again, to start off, sinoatrial node sends its wave over. There's a slight delay by the time I get over to the AV node. AV node now travels down this way, down the bundle of His, down the bundle branches, left and right bundle branches, and then wraps around. Okay. So that conduction pathway, yes?
Can you do that one more time?
So if I were to, again, for lack of a better analogy, kind of light a match right here, this would travel down this pathway. Okay. It doesn't all fire at the same time. We want it to work like a fuse. Now the reason we want that to take place is because we want the atria to contract first. Contract atria first, and we'll go over this muscle action that's taking place. Atria want to contract first while the ventricles relax, and as that pathway travels down the interventricular septum, then the atria are going to relax, and then the ventricles are going to squeeze. So that delay from the atria to the ventricles is absolutely necessary in order to allow ejection of blood that's contained within the atria to push down into the ventricles. We want enough time for that to take place. So there's that reason that there's a distance between the SA node as well as the AV node. Now looking at each of these, like I said, the SA node fires at 100 beats per minute. And if a block were to take place in the SA node, the AV node can fire, and that fires at about 60 to 80 beats per minute. If the AV node were to be blocked, the SA node doesn't fire, the AV node doesn't fire, if you were to cut at any point right there, you would see that the bundle of His would also fire as well. And that's about 40 to about 60 beats per minute. And then lastly, the Purkinje fibers beat at 20 to 40 beats per minute. The Purkinje fibers down by the ventricles. Again, if you were to cut all that off, and the pacemaker cells at the SA node can't communicate down, the AV node can't communicate down, the bundle of His can't communicate down, that means the Purkinje fibers can still beat on their own, but at 20 to 40 beats per minute. So at any point of a block down that conduction pathway, there's still a fail-safe. SA node block, AV node will fire at 60 to 80 beats per minute. If the AV node stops and there's some disruption taking place, we know that the ventricles can still contract and keep blood pumping out of the ventricles. And I'll talk about those blocks later on. Now, what that means, and typically they would see if, let's say, the SA node isn't firing anymore, that means what they can do is they can actually install a pacemaker. And a pacemaker is something that basically is battery operated, and it sends a wire over to that general area. And that's going to stimulate. All it does is give a minor shock that creates the depolarization that results in depolarization taking place. And as long as you can get that signal to start in that area, then it'll travel down and move down the conduction pathway. And all that is, like I said, all it is just an electrical wire into that area. It's probably a lot more complicated than that. But that's going to start that, jumpstart those cells to beat at their own. Another things that they're also doing with these pacemakers is they're trying to find something a little less invasive. And so they're working on what are called bio-pacemaker cells. That means they can take cardiac stem cells. If they have your cardiac stem cells, they can take those stem cells and basically perform some sort of form of genetic expression to create the pacemaker current. And what that involves for that pacemaker current is something that will exhibit that autorhythmicity from resting membrane potential that will lead us to threshold. So really what that comes down to are the expression of those leaky sodium channels. If they can get them embedded around that cell, what am I looking at here? If I can get those, in each of these cells, I can get those expressed leaky sodium channels, they will exhibit that pacemaker potential and then therefore exhibit that autorhythmicity. Again, very simplified version of what they're doing, but much more complicated than that. But they can just basically inject your own cells to become pacemaker cells. That's what it comes down to. What questions do we have about the conduction system, the pacemaker cells and the conduction system? Everybody good? So now that we've talked about the pacemaker cells, the non-contractile autorhythmic cells, let's take a look at the action potential generated by our cardiac muscle cells, our myocardiocytes. Entirely, well, not entirely, but a different action potential of what you saw earlier. The resting membrane potential will start off at -90. And the action potential is going to basically be working the same way that you saw earlier. You'll see the rush of sodium causing depolarization, and then you're going to see repolarization due to potassium. That's not new. We've talked about that already. But what's going to create a prolonged depolarization is due to calcium. The movement of calcium. Just like you saw in our pacemaker cells, calcium is going to serve to prolong that contraction. And let's take a look and see what that looks like. Here you can see the different phases. We're starting off at -90. -90 is when we're going to see sodium, voltage-gated sodium channels open. You're going to see that rush. That's going to take us from -90 all the way up to about +20. And for the purposes of the test, I'm not going to have you guys memorize +20 or how high it goes up. Really, I just want you guys to get the idea that this is the depolarization. This is phase number 1 of the depolarization for a cardiac muscle cell. Once we get to the very top, our voltage-gated sodium channels will close. And then our voltage-gated potassium channels will start to open at this point. Voltage-gated, close, potassium opens. Now we know in a cell, when sodium rushes in, that's going to depolarize, hence bring us that upswing. We know that the other side, when we get potassium rushing out, would be repolarization. Positive ion leaving a positive interior, that's going to make it more negative on the inside. Now what's taking place right up at the middle is calcium. So at this point, right when our voltage-gated sodium channels close, we're going to now open up our voltage-gated calcium channels. So while at that point where you're going to have some potassium leaving, which would repolarize, you're going to also have calcium entering right at the same time. So I've got a positive ion moving out, I got a positive ion moving in. So while potassium efflux is taking place, we get calcium influx. And that should keep us right at that same membrane potential. So therefore, there's no changes taking place from this point up to this point. We keep that cell, that cardiac muscle cell, at a depolarized state. So the length of depolarization is this entire green section right here. We'll talk about why the depolarization is necessary in just a second. Right when we get up to this point, we'll see those calcium channels start to close out, and then continued potassium efflux. So potassium continues to flow out, therefore contributing to the repolarization state. So all in all, what am I getting out of this? I'm getting an extended depolarization state. Everything from this point all the way up to this point. That gives us one thing on here. The next line is to talk about the refractory period. Now, if you remember from our neurophysiology lecture, the refractory period is at the time at which our cardiac, or in our neurons can't fire if there's a second stimulus. Okay. That should mean that if the SA node fires, and then all the cardiac muscle cells nearby fire as well, that means if another one were to fire again for some reason, if we're to fire again, this cardiac muscle cell won't fire a second time because it's already firing. It's already maintained its depolarization due to calcium. So at no point, at this point, at this point, at this point, or at that point, will that cardiac muscle fiber fire again. Why? Because it's already contracting. And it's going to maintain that contraction right at that point in time. So how long is that length of contraction? It takes place from the start of depolarization. We get up to this point right here, and now we're going to maintain that long depolarization state, leading to this whole state of contraction. Now think about this for a second, because this is going to show you the action potential that's taking place for that same cardiac muscle cell. Did you guys find this slide?
Yeah.
Now what that's showing you is this extended phase right here. So during this period from over here, from at 0 seconds, all the way out to about 170 seconds. This entire point is the length, and you can see what's taking place for the purple line representing the tension that takes place with the muscle cell. So what's taking place at the start of depolarization of the nearby, of the muscle cells? They start the contraction, and as they continually contract, they're going to start using those muscle bridges. The myosin heads are going to start pulling against each other and generating enough tension. So it wants to continually increase in the amount of tension up to the point that we get to the very peak. And at that peak is the peak tension development, right at that point. Once we reach repolarization, which takes place from this point on, is when the muscle starts to relax and those bridges start to relax on themselves and the tension decreases over and over again. So what this comes out to, and what you learn in our neurophysiology lecture, in order to elicit this muscle tension, at the very bottom, we had to have a whole series of action potentials that kept taking place. This is what we called our tetanus. And tetanus results in continued contractions up until the point that we can continue, up to the point we get to the very top. And then, once we decrease the number of action potentials, then we start to see tension decrease. And that was my example that I used earlier when I used the example of the bicep curl. We want to be able to generate enough tension all throughout the length of that state of depolarization, which we're seeing over here, in order to continually generate tension. Why are we doing that? If I were to take a look at a muscle cell and I had one single action potential, that would mean a cell just does this and that's it. That's not what we want. We want to be able to use the heart. The cardiac muscle cells want to be able to squeeze and maintain squeezing. And maintain squeezing for this entire duration in order to develop enough tension. Get those muscle cells to continually tighten up and squeeze enough to be able to push blood from one chamber to the next. Whether we're moving from the atria to the ventricles, or from the ventricles to the aorta or the pulmonary trunk. In essence, what we're going to be seeing the muscle do is we're going to want to be able to squeeze long enough to generate enough tension to overcome the pressure in the next chamber. We'll talk about the pressures, and I believe we'll talk more about that maybe at the end of today or possibly Thursday. We want to be able to generate enough pressure to be able to push blood over to the next chamber. You'll see the length of contraction. And the term contraction is also referred to as systole. I've got other slides that will be using that term, but when you see the term systole, that is contraction. The other state, everything else around here, up until the point we get to the next depolarization, this whole state right here is called, well, it's relaxation. But the technical term for it is called diastole. And now, why do we want an extended period of time for relaxation? If we can answer the question of why do we want a long contraction, was to generate enough tension to be able to raise the pressure in that chamber and get it to squeeze out to the next. And the example I can use is a toothpaste tube. If I want to be able to squeeze that toothpaste out of that toothpaste tube, I got to generate enough pressure on the inside to be able to exceed the pressure on the outside air pressure. So once I'm able to squeeze, now that toothpaste is able to move into the airspace or whatever I'm trying to dump it out into. Now, what I'm going to do next after the end of contraction is I'm going to relax. Because now if I can relax that, I guess toothpaste doesn't work this way, but if you're to relax anything else, what that's going to allow the atria or the ventricles to do, depending on which tissue we're looking at, we're going to allow that to relax. And during that relaxation phase, we're going to have the prior chamber that's going to undergo squeezing that will allow that chamber to fill up as well. I know I'm starting to add a lot more. I'm adding the atria and ventricles, but really I just want you guys to get the idea. Contraction for that muscle, and then relaxation. There's a pattern that cell is going to undergo contraction to relaxation, contraction to relaxation. And that's going to give us enough time for filling or emptying. So with that, what questions do we have about the cardiac muscle action potential?
So this one was cardiac, the other one was pacemaker.
Yeah,
right?
pacemaker for the pacemaker exhibits autorhythmicity. This cardiac muscle action potential, even though it could beat on, it can beat on its own, it's mostly geared for this extended depolarization. And all of it comes down to is where that calcium is involved. In our prior, calcium creates this huge depolarization curve in the very beginning. Sodium is first initially involved, then calcium, then potassium. In our muscle, sodium, voltage-gated sodium channels, voltage-gated calcium channels, then potassium. So be able to, when you're doing your studying, is to think about the movement of the ions and what that's going to do for our membrane, the changes in membrane potential. And that should help explain. And all that stuff that I just drew out here, the movement, think about the movement, think about the potential, the diffusion gradient, the concentration gradient from extracellular to intracellular, and that will help play this all out for you. So why do I bring this up? This whole slide of cardioplegia. Cardioplegia is basically, by breaking up the terms, cardio, meaning heart, plegia, like paralysis, is all talking about how we stop the heart. Now you might be thinking, why do we want to stop the heart? Well, one of the reasons they want to stop the heart if they're going to perform any open heart surgery. Imagine if you were a surgeon trying to perform surgery on a heart that's beating in front of you. Not really going to be the most optimal way. The proper way is to actually stop the heart. And the way they do that, and I've got a video that talks about the organ care system, specifically for the heart. What they're trying to do in cardioplegia is to get it to stop beating. And the way they can do that is to basically cool down the heart itself. They want to, and why would they want to cool down the heart? Again, it's related to temperature. If you cool a tissue down, that's going to slow the movement of ions. Remember what reducing temperature is going to do. If I'm going to heat something up, that's going to get our ions to move even faster. We're going to increase the amount of kinetic energy. If I cool something down, it's going to slow that down and reduce the amount of kinetic energy. So as ions start to move slower, that technically means that the heart is going to start beating a little bit slower. But what they're also going to be doing is they're going to actually inject a solution that's high in potassium. Typically they use potassium chloride. And they can inject that into the heart, or they can inject it, they can bathe the heart in potassium chloride, and all that's doing for the cells is increase the amount of potassium extracellularly. Now we have to think about what that's going to do. Inside the cell, we already know that there's already a high potassium on the inside. And there should be low potassium on the outside. So with that, there should be an established concentration gradient that pushes outward due to the concentration gradient out. And then the voltage gradient should be inward. So this is concentration gradient, this is voltage gradient going inwards. And again, if you don't remember that, revisit that on your notes. Voltage gradient, because we have a positive ion, it's negative on the inside. Here it's about negative 90 millivolts. We've got a positive ion wanting to go in. We've got potassium wanting to go out. Now, if I increase the amount of potassium on the outside, that's going to decrease the concentration gradient of potassium going out. What that ultimately means, if I got less of a drive to push potassium out, I've got more positive ions on the inside. Hence, what that's going to do for the membrane potential, the resting membrane potential, if it's negative 90, that's going to bring us closer to threshold. So that's going to mean it's going to be more likely to fire. And that means once that heart undergoes that depolarization, it's going to take that one last beat, and then, because I've got more sodium channels activated, it's going to be deactivated, it's going to be inactivated at that point in time. It can't fire again. So it's going to undergo one final beat, and then it's going to stop. So this is our effective way of stopping the heart at that point in time, right after its last beat. So while they stop this, they're not going to just stop it without doing anything else. They're going to now hook up prior to all this, they'll hook up a cardiopulmonary bypass. And basically what it is the heart-lung machine. It's going to take the output of the heart, okay, which would be the aorta, and then they're going to want to also take the output of all the, they'll want to take all the input, mainly our vena cavas, that are returning blood from the systemic circuit, feed that into our bypass machine, pump it back out, be able to oxygenate it, and put it back into the aorta. So this machine is doing what the heart and the lungs are supposed to be doing, pump blood through and oxygenate it in the lungs, therefore bypassing the heart. Once you bypass the heart, you can now cool down the tissue, you can do your cold infusion into the arteries to slow it down, and then inject your potassium to give it its final beat. And at that point in time, that heart is now stable, it's not beating anymore. You've stopped the contractile properties, and you can now operate on it. You can cut it open, you can install some valves on there, if any of them are malfunctioning. You can do a heart transplant if you need to, move that malfunctioning heart over, and bring a new heart in. And when you look at heart transplants, all it is plumbing. All they're doing is attaching the vena cavas back in, they're attaching the pulmonary trunk back in, attach the aorta back in, and effectively, that's really it. You're going to undo. You're going to remove the potassium on the outside. You're going to start to heat up the tissue. You're going to start to allow blood to flow back in, and eventually the heart starts working back again. What does that look like? Well, I've got another video. I got a video for you that's going to go over the next. Please tell me it's going to work. Yes, great. Not entirely gory or anything. You're just going to see a heart beating. This is what's called the what's called Heart in a Box, and it's made by a company called Transmedics. Now, this video is back in, I believe, 2012. So at this time, they were doing research on ways to improve the reliability of transporting a heart over from a donor to a recipient. So as you know, sometimes let's say somebody ends up donating the heart, something happens, they're able to strip the organs. You've got a heart that is taken out of a body, and you've got to be able to transport that about 4 or 5 hours, 6 hours over to the next destination and implant that into the recipient in order to make sure that that heart still stays alive. So conventionally, what do they do for heart transplant? They put that heart in ice, and they can slow it down, and typically that should be long enough to be able to get to the point where they can perform the transplant. Now, what this machine does is, again, a company called Transmedics created this machine where you hook up a heart to a huge machine. It's going to feed blood into the system. It's going to keep the heart pumping. Normally, you don't want to transport a beating heart because obviously you don't have enough oxygen, you don't have enough glucose, you don't have any metabolic substrates. That tissue will die on its own. Here, we just want to keep the heart alive by providing enough blood for it. Let me just play the video for you guys. You don't have to watch the entire 9 minutes. Let's see if I can get the audio up and running.
We've all seen it on TV. When a donor heart becomes available, medical teams must move quickly. The organ is removed and preserved by placing it in ice.
No, that's not the one I wanted.
In a cooler full of ice. Here at the United States,
You don't recognize it? This is the very front of UCLA.
Advances in the techniques and technology used, there's one aspect of the process that has pretty much remained the same. Yep. Getting the heart from point A to point B still involves putting it in a cooler full of ice. Here at the UCLA Medical Center, they're testing out a new device that will revolutionize how organs get transported and make a cooler a thing of the past, at least for organ transport. The machine is called the Organ Care System, and it could double the amount of time a heart can remain outside of a body.
A human organ has never been kept alive outside of a human body until this machine became a clinical reality.
Dr. Abbas Ardahali is the head of UCLA's heart and lung transplant program.
It makes intuitive sense to a layperson to say, instead of having my heart on ice, I want it to be warm, I want it to be beating.
For some of his patients, like 44-year-old Sandra Aguilar, this clinical trial isn't just an opportunity to test a cool new device. It's a second chance at life.
También ustedes, y
Maybe
tú
it's skip the
sabes
sappy
que
stuff.
tú y tu hermana son mi pedazo de corazón.
Next. I'm not harmless or anything, I just want to It's a little corny, she's like, you are the heart of my life. All right.
Pues mi vida, se puede decir, ha cambiado al
Good,
100%.
decreased quality of life,
Ya no puede trabajar.
can't work.
Organ Care System, how have they responded to that?
Once they fully understand the concept, they say, so why didn't you guys come up with this sooner?
Today, 8 out of 10 available donor hearts never make it to a patient in need due to a variety of factors, including the distance between hospitals. This device could potentially expand the window of opportunity for transplants while preserving the hearts in a more optimal environment.
We feel very comfortable or confident in the organ care system's ability to at least double the heart transplant volume over the next 5 years.
Waleed Hassanein is the founder and CEO of Transmedics, the company that came up with the organ care system. His eureka moment came while training to be a cardiac surgeon.
The same organ that I'm supposed to be dedicating 10 or 11 years of my life to train how to protect. But when it comes to transplant, which is the pinnacle of cardiac surgery, the heart is just thrown in a plastic bag filled and stored on ice. That was the first revelation that there has to be something better.
Waleed hopes that one day, learning how to use this device will become part of the standard training for surgeons. But for now, the company operates a lab where doctors practice using the organ care system with the closest thing to human hearts, pig hearts. Hopefully they're gonna let me handle the hearts. I don't know if they're gonna trust me. I'm no surgeon, but I've dissected my fair share of animals with my biology background, so I'm gonna do what I can to be able to poke that thing and feel a beating heart in the machine. Once removed from the donor's body, the heart is quickly connected to the device through the aorta. This is seriously just one of the most amazing things I've ever seen, you guys. There is a heart beating right in front of me. It continues to receive warm blood, as well as a proprietary formula of nutrients. Conceivably, how long could you keep a heart beating on this?
If you can renew the pool,
blood?
it can go as long as you would like.
Tamara Hayal, the VP of Clinical Development at Transmedics, showed me how the device allows clinicians to monitor the heart's rhythm and other vital signs every step of the way. So this thing lets you know that everything is going okay?
Absolutely.
And what happens if something goes wrong?
You'll have detection here and alarms that are going on to tell you that you're out of range, and you can immediately control it. You can change pump flow, you can navigate into menus to change the settings to actually treat this heart. So you have a very comprehensive analysis of how this heart is behaving, and you have a chance to improve that behavior if needed and when needed.
So I have two questions for you. One, how fragile is the heart? And two, can I touch it?
One, it's not very fragile. Two, yes, you can touch it, but it's not the norm during clinical practice.
Okay, not the norm? We're gonna make an
by all
exception.
means, you can touch it. This is a training part today, so by all means, if you want to feel the ventricle pulsating, right side and left side, and you can feel the vessel itself, see the pressure in the vessel.
It's warm.
It's warm, it's beating, it's functioning. It's just functioning as if it's in your body.
And it's not just hearts which can be transported on this system. There's a similar study currently underway with lungs, which has yielded some promising early results.
You see it opening and going up and down?
So far, it's cut the complication rate by more than 60%.
We'll save the lungs for next time. Now that you see how that's working. What we described so far, the pacemaker cells, the action potential is related to the pacemakers, as we mentioned, the SA node, over to the AV node, down the conduction system, down the interventricular septum, the Purkinje fibers wrapping around. Now that we described the muscle cells and how they perform, we know that we get an extended depolarization for those muscle cells. We know they want to contract, and they want to continually contract to be able to generate enough muscle tension. Now, what we want to start to look at next is combine all those cells, all those muscle cells, and now that we understand the action potentials, we want to see how those muscles work together as a tissue. And if you were to take a look at the muscle layers surrounding the heart, you can see that they're actually all bound together. There's a pattern to them. And that pattern is called a functional syncytium. And here you can see the term sync for synchronization and idiom, whatever that means. Now, the way these cells work, these muscle cells, you can see where they wrap around the most. You can see that they wrap around each of the atria. And then you can see this next band of heart cells that wrap around the ventricles. And what you can imagine, when these cells fire, or when these tissues, when all the individual cells fire at the same time due to the pacemaker cells, you'll see that these will be the first to beat, and then the ventricles are going to be the last to beat. And that's all due to the delay as that pathway travels down that conduction pathway. So let me draw in yellow here I can draw the SA node. I can then move down the AV node and draw that pattern up on this image. So you can see where this pathway is leading. We want first the atria to contract, then we want that pathway to travel down to the ventricles, we want the squeezing to take place down over at the bottom by the apex, and then towards the lateral sides. And the purpose, like I said, the first portion of that muscle syncytium is the atrial muscles. You want the atrial muscles to fire first. That's going to allow enough squeezing of the atria to make it down to the next chamber, which are going to be our ventricles. So squeeze here first, relax the ventricular muscles. That will allow contraction, emptying of the atria, and then filling of the ventricles. Then the next portion, once the atria are going to relax, the ventricles start. So once that's going to make that conduction pathway makes it all the way down, we're going to get the ventricles to squeeze first. You'll see the first place to squeeze first is down at the very bottom of the interventricular septum, the apex of the heart. We want the squeezing to start from the bottom in order to squeeze towards the lateral sides. Why do we want that to first squeeze out? Take a look at where the outputs are. Okay. The pulmonary valve as well as the aorta. And this isn't, aorta is back over, okay, I'm sorry, I'm looking at the wrong way. Aorta back over here, so this actually technically goes back underneath. And you see the outputs are towards the top of the heart itself. So we want to squeeze from the bottom and squeeze towards the very top. It's kind of like toothpaste. If you ever see how people squeeze toothpaste, a majority of people that squeeze toothpaste squeeze right in the middle of the tube, right? And what does that do for you? Especially when you're like halfway through the toothpaste. You're just squeezing toothpaste down to the bottom of the tube. That's not the way to do it. You gotta roll it from the bottom and squeeze all the way up, right? So that's exactly what we're doing with the ventricles. Start from the bottom, squeeze all the way up to be able to eject blood efficiently out to the top of the heart. Again, the output being the aorta and the pulmonary trunk. So now that you see the patterns, atria first, then ventricles, we want to break down and take a look at what's taking place, because there's actually 3 different phases that take place. We want to see the atrial contraction take place first. While the ventricles are relaxed, we want the atria to squeeze. That's going to give us enough time to empty out the atria, to squeeze blood and allow filling of the ventricles. Now, when the next phase, as we move down the conduction pathway, we want the atria to relax, and we know when it's relaxing, it's starting to repolarize. We want to depolarize and cause contraction of the ventricles. And as the ventricles, you're going to see there's different phases of ventricular, and it's not called ventricular ejection, it's called ventricular contraction. The first part, just like toothpaste, you want to be able to generate enough tension to build up pressure on that liquid on the inside. You want it to get enough pressure to be able to build up so that by the time you build up enough pressure, it's going to be exceeding the pressure in the aorta as well as the pulmonary trunk. Okay? That pressure is the diastolic pressure. And I'll go into that especially in our vessel lecture. There's a certain amount of pressure because there's already fluid filled up in here. So I want to be able to generate enough tension first, and then when I'm squeezing even harder, that's going to push blood finally to escape. So if I were to give you an example, that would be the door. And that door, imagine this were the heart, and out there would be the aorta. The aorta has enough blood in there, there's enough pressure in there that's about 80 millimeters of mercury. And that, just think of that as a force. We'll just say, for lack of better terms, 80 newtons of force pressing against that door. In order for us to be able to, we're getting filled up by the atria, we're now filling up in here, and we're starting to contract the room. We're going to start contracting the room enough to generate tension so that we start building up pressure. We start off at about 30, 40, 50 millimeters of pressure. We keep generating enough pressure. There's no changes in the volume of blood. It's still all in here. We're just trying to contract the walls enough that eventually we get up to about 79, 80 millimeters of mercury. And the pressure outside is now pressure on, equalized to the pressure on the inside. We're going to keep contracting. And eventually, when I overcome that 80 newtons of force exerted by the aorta, I can finally open up that valve and now start emptying blood out. So you'll see 2 different patterns when we talk about ventricular contraction or ventricular systole. That's going to be called the I'll go into it later. It's going to be our isovolumetric contraction and our ventricular ejection. So that's going to be our second phase. Atrial contraction, ventricular relaxation. The next phase would be a combination of atrial relaxation and ventricular contraction. And then the next phase after that will be relaxation of the atria and ventricles at the same time. So those are the three phases that I will go over on the next couple slides. Given all that, what questions do we have so far? We good? All right. Let's move on with a video. So now this is going to place it all together, because I know my hand animations weren't exactly the best representation of the heart. We've got a video for that that'll play it all for you.
The heart is a muscle about the size of your fist. It lies behind and to the left of your breastbone or sternum. The purpose of the heart is to pump blood through blood vessels, arteries, and veins to all parts of your body. The inside of the heart is divided into 4 chambers. The top 2 chambers are called the atria and are collection chambers for blood. The bottom 2 chambers are called the ventricles and receive the blood from the atria and pump it to the lungs and the body. The chambers are separated by valves, which control the direction of blood flow. There are 4 valves: tricuspid, pulmonic, mitral, and aortic. Circulation begins at the right side of the heart, where blood from the body comes to the right atrium. This blood passes to the right ventricle, where it is pumped to the lungs to receive oxygen. Once it receives oxygen, it flows to the left atrium, and then to the left ventricle, where it is pumped to the aorta and the rest of the body. On the right side of the heart, the tricuspid valve separates the right atrium and the right ventricle, allowing blood to enter the ventricle, but not flow backwards to the atrium. Blood flows through the pulmonic valve to go to the lungs. On the left side of the heart, the mitral valve separates the left atrium and the left ventricle. Blood flows from the left ventricle to the aorta through the aortic valve and to the rest of the body. Arteries carry blood with oxygen and other nutrients throughout the body. Veins take blood back to the heart, which pumps it to the lungs to be oxygenated. The heart arteries, coronary arteries, provide oxygen and nutrients to the heart muscle. The right coronary artery supplies blood to the bottom and the back of the heart. The left coronary artery splits into 2 vessels. One branch supplies blood to the front of the heart. The other branch delivers blood to the left side of the heart. An electric system transmits signals throughout the heart to control its pumping. The electrical signal starts in the sinoatrial, or SA node, which is located in the upper portion of the right atrium and is known as the natural pacemaker of the heart. The electrical signal passes down to the lower chambers of the heart via the atrioventricular, or AV node, which controls the signal so the atria contract before the ventricles. In the ventricles, pathways carry the signal throughout the muscle so that they contract at the same time to pump blood to the lungs and through the body.
So hopefully that pieces it all together. And we'll move over to the next slide, which is going to tie it all together. We want to take a look at the electrical activity that is taking place on the next slide over. So looking at this, you're going to see the autorhythmic, again, our pacemaker cells, in contrast with our cardiac muscle cells action potential. So what we're going to first look at is our SA node, which we know is going to be our pacemaker, so they'll label these P for pacemaker. Our AV node, which is also going to be consisting of our pacemakers. And if you were to look at the waveforms of the pacemakers, here you can clearly see this is the pacemaker potential, here's the pacemaker potential. Eventually we'll reach threshold, and that will result in calcium influx, potassium efflux, and back to where we started. If you were to take a look at the SA node versus the AV node, you can see that the SA node starts, the wave of depolarization is going to start off at 0 seconds, which is going to be the very first starting point. The AV node, you can see taking place about halfway through. So about 50 milliseconds difference in between the first and the second, between the SA node and the AV node. And that represents the delay. And that delay, again, is from the SA node to the AV node, allows for a good enough time so the atria can contract. That's exactly why we want that delay in place. We want to give it enough time so that the atria can fully contract before the ventricles start their depolarization. That's going to describe our SA node to AV node. The next thing is to take a look at, let me use green for this one, the SA node to the atrial muscles. So the SA node is going to be the first to fire. And that is going to trigger all the adjacent atrial muscle cells to fire as well. And again, the different waveforms you would see are atrial muscle, and you'll see our ventricular muscle. Again, rapid depolarization due to sodium, extended depolarization due to calcium, and then continued repolarization due to potassium efflux. So you can see that this pattern right here, the length, which is even more pronounced for, a little less pronounced for the atria, a little more pronounced for the ventricles. You can see that the length of the contraction for the atrial muscles versus the length of the contraction for the ventricles. Again, that what I've drawn out represents systole, the length of contraction. And again, systole is everything from depolarization to repolarization. And this should make sense. If you take a look at the atrial contraction, we just want to contract enough to be able to take that blood that's held in the atria and dump that into the ventricles. When we get into ventricular systole, you'll see ventricular systole represents a longer contraction period. Why? Because we've got more blood to move out of the ventricles. So we want an extended contraction period for that portion. And you'll see that these two sections do not overlap. So atrial contraction takes place first, atrial systole, and then ventricular systole takes place after. So not only can you see the node from SA to AV node, you can see the delay in the conduction from SA to AV node. And as this makes its way over, you'll see that SA node, atrial muscle, AV node, ventricular contraction takes place in sequential order as it moves down that conduction pathway.
You said systole is depolarization and repolarization?
The time in between depolarization and repolarization. So if I were to take you back over to this period right here, depolarization to about depolarization to about repolarization. And I know there's a little overlap with repolarization, but you can still just appreciate the fact that once you get to a certain point of repolarization is when the muscle starts to relax. So it is a little bit overlap. But systole is, for all intents and purposes, start of depolarization to the end of depolarization. Probably depolarization to repolarization. That's what I'll label it as. Way too fast. All right, so that is systole. Again, atrial systole, ventricular systole. The other thing that we can look at is the delay in between. So everything out from this point out is diastole. And we want diastole for the ventricles, ventricular diastole. There is an atrial systole and an atrial diastole. There's a ventricular systole and diastole. And all that systole means contraction, relaxation. So don't confuse the two. Ventricular diastole is for the relaxation in order to allow filling. So while this is contracting, the atrial muscles are contracting, that's continued filling of the ventricles. And then once we get to repolarization, we're going to relax the atrial muscles. And then once the AV node makes its way down, or the electrical pathway makes its way down, all the way down to the Purkinje fibers. Then we start to see the start of ventricular contraction, or ventricular systole. And this is going to empty blood out to the outputs of the heart, aorta as well as the pulmonary trunk. And then from that point on, we relax from here to the very end before we start to initiate the next beat. You can see this period is relaxation. So we can divide that up. From ventricular relaxation, ventricular contraction, and then back to ventricular relaxation. So 3 periods that we can see from the very start of the cycle to the very end of the cycle. And then later on, we'll break this ventricular contraction down, just like I described earlier. Now that we've been able to piece together the action potentials, let me ask you guys, what questions do you guys have? Everybody good? So what we're looking at, just to sum up everything that we've looked at, is one cardiac cycle. And the length of the cardiac cycle, again, really a good chunk of it is dependent on that pacemaker potential. When that SA node fires, you're going to see everything else will fire. Atrial muscles, the AV node, as it makes its way down, you're lighting off that fuse that will make its way all the way down and cause the muscles to contract. All of this is one cardiac cycle. From the point that we start, that SA node starts the firing, up to the point that conduction travels all the way down to the Purkinje fibers, and that SA node fires again approximately 0.6 seconds later. So every time that SA node fires, that starts the heartbeat. So as I said, this heart doesn't have any central nervous system stimulation taking place. It can fire on its own, and the SA node fires at 100 beats per minute. But that doesn't mean that this heart is not under autonomic regulation. It is. So in fact, our hearts aren't beating at 100 beats per minute all the time. They're continually adjusting based on the amount of sympathetic output and parasympathetic output. So this is going to tie back into our lecture that we looked at when we looked at our peripheral nervous system lecture, specifically autonomic regulation. So looking at the pathways, we know that the medulla oblongata is going to contain nuclei that are going to be responsible. It's going to be receiving sensory information about, which I'll talk about in the next slide over. It's going to be receiving sensory information about the state of our metabolic needs. It's going to measure the heart. It's going to measure the output, the flow of blood through the heart. It's going to measure blood pressure. And all of that is feeding into the medulla oblongata, which are going to contain 2 specific nuclei. We're going to have a cardio-excitatory area, and we're going to have a cardio-inhibitory area. And those areas are either going to speed up the heart or slow down the heart. And if we take a look at the, let's start off with the cardio-inhibitory, so parasympathetic output. Parasympathetic output that starts from the cardio-inhibitory area, we're going to see a dorsal motor nuclei of the vagus nerve travel out that nuclei of the cardio-inhibitory center, travel out. You're going to have a long, this is a pre-ganglion neuron, a long pre-ganglion neuron, axon, that's traveling all the way to the ganglion close to the heart. So there's going to be our ganglion, our parasympathetic ganglion that's close to the target tissues, and we're going to have our short post-ganglion axons that travel from the ganglion out to the SA node and the AV node. And that should make sense because these areas, the SA node and the AV node, the SA node is the pacemaker. It's the one saying we beat at 100 beats per minute. So this vagus nerve is the one, cranial nerve 10, travels all the way out and innervates those 2 pacemaker areas. If we're to take a look at the cardiosympathetic, or the cardio-excitatory area, again, that's going to travel out from that nuclei. That's going to innervate the spinal nerve area that innervates the heart. We're going to take a look at where this starts off at. It's going to start off at the lateral gray horn, not the dorsal gray horn, because the dorsal gray horn, the ventral gray horn is going to be somatic. These are going to be our autonomic areas. the lateral gray horn all the way out the sympathetic trunk ganglion. Sympathetic trunk ganglion out is going to be our long postganglion axons that terminate on the SA node, the AV node, as well as the ventricular muscles. So you'll note that the effects of that, if we get excitation of the sympathetic pathways to the heart, we're going to stimulate a higher depolarization rate, meaning we're going to increase the pacemaker potential of the SA node and the AV node to fire at an even faster rate. Now, the parasympathetic output does the exact opposite, where it's going to innervate the SA node and AV node, and parasympathetic is going to slow that down. So remember, parasympathetic being rest and digest. So we want to bring that back down from 100 beats per minute down to about 60 to 70 beats per minute. The only difference between the two is that the sympathetic output targets ventricular muscle mass, and something that we'll learn later, it'll also affect the vessels as well, the veins. All of this, the adjustments that are taking place on the heart rate at the SA node as well as the AV node, the sympathetic pathways, and all of this is written out for you guys on the next slide over. Sympathetic output, you would basically know that the postganglion neuron that targets the heart muscle are going to be secreting what neurotransmitter? Again, sympathetic pathway, postganglion neuron is secreting either epinephrine or acetylcholine. Which one is it? Which one? Acetylcholine? Remember, these are going to be adrenergic receptors on the SA node and AV node. So if they're adrenergic, they're going to be secreting epinephrine. So sympathetic pathways, remember, sympathetic pathways, epinephrine. If I want the heart to beat faster, remember what I mentioned about Pulp Fiction. They took that needle filled with epinephrine, adrenaline, and stabbed it into her heart. And that caused the heart to start beating faster. So the neurotransmitter that's released is epinephrine. If you were to take a look at the parasympathetic pathways, the postganglion neurons are going to secrete acetylcholine. So depending on what you actually just sprinkle on the SA node and AV node, if you sprinkle epinephrine, that's going to raise the heart rate. If you sprinkle acetylcholine on the AV node, that's going to slow it back down. And tons of these studies have basically done that with hearts. What things can we put in that comes in that will speed up the heart? One of the things that I mentioned earlier, SA node fires at 100 beats per minute. And they know that, because one of the earlier studies that they used to do was they used to cut the vagus nerve on dogs. And by cutting the vagus nerve, we know that's the parasympathetic output to the heart. And intrinsically, what happens to the heart rate for dogs? It was 70, brings it right back up to about 100. So cutting off any of what we call vagal tone, so the vagus nerves provides vagal tone, which decreases the heart rate back from 100 beats per minute down to about 60 to 70 beats per minute. So what we're looking at, the set point, we're starting off at 100 beats per minute. To bring it down to about 60 beats per minute is vagal tone, or parasympathetic output. To raise the heart rate up to about 200 beats per minute would require sympathetic output. That's epinephrine. So right now, all of us are sitting presumably with a heart rate between 60 to 80 beats per minute. You do have vagal tone. That's your parasympathetic output saying you're resting, you're digesting. We don't need you at 100 beats per minute, so let's slow that back down to 60 beats per minute. Now, let's say something were to happen right now, knock on wood, but all of a sudden it's going to cause us to fight or flight. We're going to have to get up and start running. So what's going to happen? Parasympathetic output is coming in, and it's already working on the heart. Event happens. Now we're going to turn off the parasympathetic output. That's going to raise us back to 100 beats per minute. And then sympathetic output starts to increase to raise us back to a higher rate. So what that means is that your heart is undergoing a certain amount of sympathetic output and a certain amount of parasympathetic output, and it's working together to balance your heart rate at a resting heart rate of about 60 to 70 beats per minute. It's not until you want to raise it back up to about 100 beats per minute that you turn off the amount of vagal tone. And depending on your stress levels, depending on the amount of cortisol, depending on also how well your heart efficiently pumps blood, it's going to be dependent on what your beats per minute is, or your heart rate is at intrinsically. So the next slide over, all of this that we've been talking about is the efferent pathway. This is the efferent pathway from the medulla oblongata out to the heart. But what we do need to talk about is the sensory information. So we've talked about the first portion. Now we'll talk about what sensory information. And here you can see our integration center, the integrative function of the whole sensory, integrative, motor pathways. Motor pathways we already described earlier. Cardio accelerator is going to increase the rate of depolarization of the SA node. It's going to also increase contractility of the ventricles. If we look at parasympathetic output only decreases the depolarization of the SA node as well as the AV node. So decreases heart rate. We've already talked about that. The sensory input that comes in and feeds the medulla oblongata, we want to be able to receive information about the amount of blood that's flowing through the system. So the areas that we probably would want to stick those sensors in to help us adjust the amount of blood flowing to those areas would probably be the aorta. Probably makes sense because that's our direct output for systemic. This is our direct systemic output, and probably some other receptors leading up to the brain itself. So the carotid body's located. That would probably be good areas. So where do we see these baroreceptors? Baroreceptors detect the amount of pressure, the blood pressure in those areas. And by the amount of pressure, we can also determine the amount of volume and flow of blood to those areas. So it's all related. Volume, flow is all related to pressure, something we'll learn in our respiratory lecture. And actually this lecture as well. So we want to make sure that the pressure is appropriate, because that pressure is going to be important for blood flow throughout the systemic circuit, which includes blood going up to the brain. So we want to make sure we got enough pressure, enough blood flow to the critical organs. So naturally those baroreceptors are located in the aorta as well as the carotid bodies. What other information leads into the medulla oblongata from other brain centers? You got your cerebral cortex. Your primary motor cortex is saying, I'm doing a lot of movement right now, so you probably want to ramp up the heart a little bit. That's going to lead into the medulla oblongata, specifically into the cardioacceleratory areas. Your limbic system is going to be involved in emotions. You start to get really emotional, start getting a little angry, you're going to start raising your heart rate back up. Looking at the other sensory receptors that are located. Baroreceptors, which we already mentioned, which detect pressure in those areas, aortic arch as well as the carotid bodies. Chemoreceptors are also going to tell us the state of chemicals in the blood. Is the CO2 too high? Is our oxygen levels low? Do I need to start increasing blood flow through the heart? Am I starting to detect the presence of excessive hydrogen ions? Maybe increased acids are forming in the blood, decreasing the pH of the blood.
Do like drugs affect chemoreceptors?
Down the line, yeah. There can be, looking at those pathways can actually start to, keep in mind that those pathways start to affect the amount of catecholamines in the system, so the amount of epinephrine. So those things could actually start exciting brain activity that could tell the cardiovascular regions to start ramping up. They might not directly affect the medulla oblongata, and that I don't know, but they'll certainly have an effect somewhere up in the central nervous system. Because it's a central nervous stimulant. If you look at speed, which is an amphetamine, will stimulate central nervous system, which will then feed into the cardiovascular center. That targets our chemoreceptors, so any hydrogen ions that would lower the pH, like metabolic acidosis, the production of lactic acid. Right? Those types of receptors will say you may want to speed up the heart again. The other thing on here that you'll see, proprioceptors, things involved in motion. So any movements that take place, again, feed into the cardiovascular region to say ramp up, you gotta increase your blood flow, therefore you need to increase the heart rate. So the other things to look at in this table, as you know, I really like my tables to help kind of sum up all that information. You can see the SA node, AV node, and ventricular conduction pathway are all going to be related to the pacemakers. The atrial muscle down to ventricular muscle are going to be our muscle cells. And this is the effect of parasympathetic and sympathetic stimulation. So we know what's going to happen with vagal tone. Vagal tone is going to decrease the SA node and AV node. It's going to, parasympathetic output, even though I didn't say it doesn't really have an effect on muscle, it has a slight effect on the atrial muscles, so don't worry about that. All in all, if I were to ask you about vagal tone, predominantly going to affect the pacemaker cells. Sympathetic output is going to increase the rate of depolarization of the SA node and AV node, and it's also going to increase the strength of the contraction. So that sprinkling of epinephrine and norepinephrine on the heart itself will affect the intrinsic rate of depolarization in the pacemaker cells, as well as the strength of the contraction. Because that's going to contribute to a concept that we'll talk about at the end of, on the beginning of Thursday. Not only do I want my heart to beat faster, I also want the heart to beat stronger. I want to increase the amount of blood that's ejected in that heartbeat. And that example is going to be both the combination of the heart rate as well as the amount of blood ejected per beat is going to be a concept we're going to call cardiac output. It's how much blood is flowing through the heart itself. And during exercise, you'll feel your heart thumping out of your chest. Not only is it beating faster, but it's beating stronger. We want to be able to increase the amount of blood flow. That's our cardiac output. Again, something we'll save later on. Summing that all up, increased contraction of the atrial as well as the ventricular muscle due to sympathetic output. And that, again, can be visualized based on this right here. Sympathetic output to the muscle cells. So what we've again talked about the entire, in the last couple slides over, are the action potentials that are generated in the heart. Out of looking at the amount of mass in the heart, the SA node being a couple millimeters squared in surface, in volume, and the AV node and the other branches being a couple millimeters squared or cubed in volume as well. The majority of all the cells in the heart are mostly muscle cells. Wouldn't you agree? Yeah? Okay. So mostly muscle cells, almost all entirely muscle cells. If you were to measure the activity going on, and again, going back to our action potential for the cardiac muscle cells, you would probably see that the most activity that would be measured would be taking place with the cardiac muscle cells. And we know that these cardiac muscle cells beat, depending on where they're located, whether the atria or the ventricles, beat at different times. So what we're going to be doing for the ECG, what exactly is the ECG? It's our electrocardiogram, also known as Okay, how many people still refer to as EKG? Probably a lot. What is it? What is EKG? It's Electrocardiogram in German. EKG, but here we call it ECG. So you may get corrected if you ever call it an EKG. It's called an ECG because we call it electrocardiogram. And what an electrocardiogram does is it just basically measures the changes in membrane potential. So if I were to measure a cell, I've got my lonely cell here, and I'm just going to measure the changes that take place from resting membrane potential. I'll notice I'll have my depolarization, I'll have my extended depolarization, and then I'm going to have my repolarization. This is measuring the changes that are taking place in terms of negative 90 millivolts. These are absolute changes that are taking place. So about 20 millivolts. We want to measure any changes when the cell is at rest. So when a cell is at rest, and here we can just draw it as a 0. When you see a heart cell and there's no changes in the membrane potential, it'll be a 0, right? And so if it stays 0 the entire time, that means the heart cell is not working, it's not beating. But that's not what's taking place. You saw in the very beginning a huge rate of change in the depolarization. So we would expect, right at the very beginning, here we see our cell at rest. Let me just draw this in. Here we see the heart at rest. And I'm just going to draw, let me draw that depolarization curve right up on top. Can you guys see that? Kind of. Okay. So here's our depolarization curve at the top. We see the biggest change taking place in the very beginning. Okay, so I'm going to start over with, which is good because then I can kind of redo what my thing looked like. I'm going to draw my depolarization curve up here at the top. Here's at negative 90. Here we are at rest. And then I'm going to draw my depolarization back down and back down over here. So this is my cardiac muscle. Let's just call this, let me just make it easy. Let's call it our ventricular muscle. Now, measuring any changes that are taking place, I can draw out on our graph, which is going to measure any changes, the rate of change of the depolarization and repolarization that's taking place during muscle contraction. So the change taking place at the initial portion, which I'm going to draw in blue, it's going to stay 0, right up at that point in time. Right here we see that large depolarization that takes place. So I see a huge change taking place. If this is depolarization, what I should see is a little blip. And then, once it reaches the very top, it slows down, and then the change from this point on, is there any changes taking place from this point at the top? No. So we should even back out. When does the next change take place? During repolarization, where you're going to see now a change in the membrane potential, so you should expect to see another blip. And then it evens out. So everything here, this is going to represent the depolarization, and this is going to represent the repolarization, which are going to represent huge changes in the membrane potential. That's exactly what an ECG does. It measures when those changes occur. So if you were going to take a look at the ventricles, you'll see a blip when you see depolarization. You see another blip with repolarization. So now let's draw that out. Let me draw what this would look like with both atria and ventricles. So the first thing you would expect to see is the blip with the atria. The atria we know, the SA node fires, the atria contract. Again, atria being, at least looking at just at the muscle cells, we want to be able to measure the muscle cells changes in the depolarization and repolarization. So we expect to see a little blip first, which is going to be atrial depolarization. And then, what would be the next blip? If I had atrial depolarization, what's the next one? Atrial repolarization is the next blip. Then, what's the next thing I should see? What contracts after the atria? Ventricles. So the next blip I should see, ventricular depolarization first, right? Then the next blip I should see, ventricular repolarization. So it should look like this. But technically our ECG doesn't look like this. It looks combined. The first bit you see is what's called our P wave. And our P wave, and all of this is described on the next slide over. Okay, I'll do it off this one. The first thing that you're going to be able to see is the first blip would be atrial depolarization. It starts off with the SA node starting, and you're going to see the waveform, the movement of our sodium ions moving through the cardiac muscle cells. You see the first portion, now that wave spreading, this is our depolarization. It's the start of contraction, which is marked in yellow. The next blip you're going to see is when the repolarization starts, which is down over here. And it's not until you get to this portion right here where the atrial relaxation finishes or starts. And atrial relaxation is repolarization. So let me go back to this portion right here. P wave marks the start of atrial depolarization. This point is where atrial repolarization takes place. At the same time, after atrial repolarization takes place, there's another thing that takes place, which is ventricular depolarization. So in essence, what this is doing, it's creating one big blip. Because not only am I getting atrial repolarization, I'm also getting ventricular depolarization. So I'm getting a lot of changes that are taking place in the atria as well as the ventricles. So that's going to be combined. First is going to be your P wave, then this whole segment right here is called the QRS. And the R wave is the biggest deflection that you see. And that's the start of ventricular depolarization. Now, the last portion, which you guys described, is ventricular repolarization, which is exactly what T is. And while you guys are all writing that down, I got that all for you right over here. Next slide. So this is going to be important because what we can do from here, why this ECG is so important, again, it's measuring the changes. Take a look, it's millivolts, changes in millivolts that's taking place. 0 when the heart hasn't started its cardiac cycle. At the start of the cardiac cycle, P represents atrial depolarization. Then we go into atrial repolarization. At the same time, ventricular depolarization takes place. The reason you don't see a separate blip is because the ventricular depolarization masks atrial repolarization. Then, that last segment would be ventricular repolarization. Those three big waveforms and all the time points in between tell us exactly the timing of the heart. The time that I can see, which is our PQ interval, if I were to note where the big segments are, and let's take a look at that on the next slide over. R is QRS. This whole segment from QRS to about T, which they actually call the ST segment. The ST segment, let me draw that up. From QRS marking the start of ventricular depolarization, all the way to about T, which starts the beginning of ventricular repolarization. That entire length right here is ventricular systole. That is the time of ventricular contraction. So that segment right there, the ST segment, is important because during ventricular contraction, especially the latter portion of ventricular contraction, signifies the amount of time that it takes for the ventricles to squeeze and empty the blood out. An ST segment is extremely important because what happens if you see somebody's ECG and the ST segment is short? That means that the ventricle did not have enough time to empty out completely before ventricular repolarization takes place. Hence why ST segment is extremely important and something that they want to look at in an ECG. If you don't have enough time to empty out the ventricles, well, you're not completely emptying out the ventricles, but if you can't get enough blood out of the ventricles, that means that's not enough blood flowing through the systemic circuit. So you've got reduced output out of the heart flowing through the ventricles, not providing enough blood. And they can see that just off a bunch of electrodes placed on your chest. The other thing that we look at is the PQ interval. PQ interval, again, between atrial, the start of atrial depolarization to the start of atrial repolarization, is the amount of time for the atrial kick. Atrial kick is atrial contraction. Can the ventricles be filled up during, if this were systole, if all this is systole, the rest is ventricular diastole. And that's the time that ventricles are relaxed. Is there enough time in the PQ interval? Is the PQ interval appropriate enough? Is it long enough to be able to fill the ventricles up? Because it's one thing if during the ST segment, can the ventricles squeeze enough blood out? But can it also be filled by the atria during atrial systole? So these are the things to consider when you're looking at an ECG. You can just measure the times because frankly, the ECG waveform is fairly standard. You've got machines that you plug them in, and they will measure what the reference intervals are based on your age, your gender. It knows what normal values are for PQ segments. It knows the length for ST segments. And especially measuring this over changes, let's say at times, let's say when, and this is the reason why they do stress tests, they do treadmill tests with ECGs. They want to see how does the heart respond, not only at rest, but also when you're performing any form of exercise. Let's say you're out and it doesn't necessarily have to be like a marathon you're running. These could be things that increase your heart rate. Does your heart operate normally with increased sympathetic output? Are there any changes in the ST segment? And any changes that deviate from normal basically mean inefficiency of the heart. Either the inability to fill completely or the inability to completely eject the proper amount of blood. So those are the things that I'd like you guys to be very familiar with. Again, looking at the ECG, measuring the rate of changes that are taking place. And all of that is broken down. When you look at the phases of the ECG, we'll do exactly this for you. They'll pinpoint the start of atrial depolarization, repolarization that's starting to take place, the conduction moving down to the Purkinje fibers, and then this huge waveform is the start of ventricular contraction or ventricular depolarization, and then this will relax at our T wave at the very end. Each of that is broken down for you, going through each of these segments taking place. So take that and make sure that you move through each of the segments to see when things are taking place. Again, systole marks the time from depolarization to repolarization. Here you can see where ventricular systole takes place. This is atrial systole. Everything else outside of this is ventricular diastole. Now we know what's happening with the muscle cells, that we've been able to sum that up. Let's take a look at a video that will, got two videos. Just give me a second, I'm trying to figure out how far we can go. 3 minutes and 45
The
seconds.
cardiac conduction system consists of the following components. The sinoatrial node, or SA node, located in the right atrium near the
Good videos I
entrance
think I
of the
posted on
superior
Canvas that
vena
you guys can
cava.
watch also in the
This is
Google
the
Drive.
natural pacemaker of the heart. It initiates all heartbeat and determines heart rate. Electrical impulses from the SA node spread throughout both atria and stimulate them to contract. The atrioventricular node, or AV node, located on the other side of the right atrium near the AV valve. The AV node serves as electrical gateway to the ventricles. It delays the passage of electrical impulses to the ventricles. This delay is to ensure that the atria have ejected all the blood into the ventricles before the ventricles contract. The AV node receives signals from the SA node and passes them onto the atrioventricular bundle, AV bundle, or bundle of His. This bundle is then divided into right and left bundle branches, which conduct the impulses toward the apex of the heart. The signals are then passed onto Purkinje fibers, turning upward and spreading throughout the ventricular myocardium. Electrical activities of the heart can be recorded in the form of electrocardiogram, ECG, or EKG. An ECG is a composite recording of all the action potentials produced by the nodes and the cells of the myocardium. Each wave or segment of the ECG corresponds to a certain event of the cardiac electrical cycle. When the atria are full of blood, the SA node fires. Electrical signals spread throughout the atria and cause them to depolarize. This is represented by the P wave on the ECG. Atrial contraction, or atrial systole, starts about 100 milliseconds after the P wave begins. The PQ segment represents the time the signals traveled from the SA node to the AV node. The QRS complex marks the firing of the AV node and represents ventricular depolarization. Q wave corresponds to depolarization of the interventricular septum. R wave is produced by depolarization of the main mass of the ventricles. S wave represents the last phase of ventricular depolarization at the base of the heart. Atrial repolarization also occurs during this time, but the signal is obscured by the large QRS complex. The ST segment reflects the plateau in the myocardial action potential. This is when the ventricles contract and pump blood. The T wave represents ventricular repolarization immediately before ventricular relaxation, or ventricular diastole. The cycle repeats itself with every heartbeat.
So now what we've talked about the electrical activity of the heart. We've talked about and this is specifically looking at the ventricle, the ventricles. We see the ventricular action potential in purple. We see the electrical activity of the heart, which you can see our ECG in the bottom. What we do need to now take a look at are the changes in pressure that take place as a result of the contraction that takes place. Now in this slide, what we can first start off with is pointing out where, and I believe I go over this later on. Oh yeah, okay, you know what? That's okay. I just want to show you how complicated it's going to get. So here you're going to be able to see the pressures taking place. This is going to be part of the diagram where, at least let me just finish up my sentence. We can start off by, in preparation for Thursday, is to note where ventricular systole takes place. So this marks ventricular systole. You can see that taking place with the ventricular action potential. We're going to want to take a look at the changes in pressure that happen in the ventricle as well as the aorta. So that whole example that I talked about, where this was the heart, that was our aortic valve, that door, and outside was the aorta, are all going to be relating the pressures that are taking place during ventricular diastole to ventricular systole, which we mark off as QRS, and then the T wave, and which we can see up on our purple line, the beginning of depolarization to the beginning of repolarization. The left ventricular pressure during ventricular diastole is close to 0, about 0 to 5 millimeters of mercury. Right at the very beginning of ventricular diastole, ventricular systole, is the marked increase in pressure. So remember what I was saying earlier is ventricular muscle walls are contracting. You see the pressure rising. Now the measurement of the aortic pressure is the back pressure exerted by the aorta on the aortic valve. So in order for us to open that door, we have to be able to generate enough pressure to exceed 80 millimeters of mercury of pressure. So what's going to happen is the pressure keeps rising. The volume doesn't change, and this graph doesn't even show you volume. That's something I'll show you in Wigger's diagram. The pressure starts to increase, keeps increasing, up to the point that the pressure now matches, the pressure in the left ventricle matches the aorta. So in that example that I mentioned earlier, you have 80 millimeters of mercury being exerted by the aorta onto the aortic valve. You have now 80 millimeters of mercury of pressure pushing up due to ventricular contraction or ventricular systole pressing up against the aortic valve as well. The second that aortic, the ventricular pressure exceeds that of the aortic pressure is where you start to see blood flow taking place. And blood flow is now being ejected. Remember, this is still continued contraction. We get blood flow being ejected out of the left ventricle. And the peak that it reaches is 120 millimeters of mercury. Up until the point, now we're at relaxation, and now the pressure starts to decrease. Decrease. And we're going to start to decrease up to the point that the aortic pressure is going to be higher than that of the ventricular pressure. So this marks the point at which the aortic valve closes. This portion, not that portion, this point right here marks where the aortic valve opens. So this whole point right here represents, from here to here, measures the amount of blood that's ejected by the ventricles when the aortic valve opens to the point it closes. You'll see pressures drop. Why? Because we're in ventricular diastole. And then pressure comes back to normal. So like I said, that's going to show you a taste. We're going to add on to this the actual pictures of the cardiac cycle, as well as pictures of ventricular volume. And we'll save that for the beginning of Thursday. So in order for you guys to get acquainted with that, review this slide, because we're going to add to that. So looking at ECG abnormalities, now that you have an idea of what's taking place in terms of the ECG, the ventricular action potentials, and the pressures, which we'll worry about later. Any deviance from our blood pressures, our heart rate should be about 60 to about 70 beats per minute. That's about resting heart rate. And looking at the terms that you probably would have been seeing at some point, bradycardia is a slow heart rate, less than 60 beats per minute. Tachycardia is a fast resting heart rate, so anything above that 100 beats per minute. Typically, things that you would see with bradycardia, if people have a slow heart rate, people will exhibit low blood flow to the rest of the systemic circuit. So people typically will feel some fatigue, feel weakness, sometimes dizziness, low blood pressure to the carotid bodies. The exception to that would be athletes. People that have a tendency to be athletic will have lower heart rates than 60 beats per minute. Why? Because the heart is it's a muscle. It can completely empty. So something that we'll explore the notion later, which is cardiac output. It's not entirely related to heart rate, but it's heart rate times stroke volume. Athletes have a tendency to be able to fill blood more in their left ventricle, and they can empty more blood in the left ventricle. So increases in stroke volume for athletes will result in a decreased heart rate. So ask your athletic friends what their heart rate is, and it wouldn't be uncommon for them to be under 60 beats per minute. Now, tachycardia, on the other hand, typically is going to be excessive sympathetic output. So if you were to measure the amount of circulating catecholamines, the amount of epinephrine, norepinephrine, going to be fairly high. So high heart rate. And typically you'll see people with fever, sometimes blood pressure drops. You're going to start to see heart rate starting to increase in order to increase the amount of cardiac output, or to at least normalize it. Other things that you can also see, typically related with high stress levels, high cortisol, hyperthyroidism, give us an indication. All of these will give us an indication as to why the heart rate might be high. It's typically due to increased sympathetic output. Many reasons, but nonetheless, terms bradycardia and tachycardia are all related to the heart rate, the intrinsic heart rate. Now, looking at the abnormalities. In the abnormalities, we'll be able to see the normal sinus rhythm. And normal sinus rhythm just lets us know that P, Q, or S, and T are exactly where they should be in a normal heart. And looking at the normal rhythm, you can see right up at the very top in this antiquated ECG tracing, you can see that the very top represents the normal rhythm. And what this helps us do is to take a look at the heart blocks. Blocks are basically things that are a form of arrhythmia, and that arrhythmia is due to the lack or the dysfunction of either the SA node or the AV node, or somewhere along down the conduction system. So looking at what a heart block is, at least the first heart block, the first degree of the heart block is prolongation of the PQ interval. So here we expect P, Q, or S, and then T is P, Q, R, S, and then T wave. PQ, prolongation of the PQ interval due to refractoriness of the conductive cells in the AV node. All that's basically saying is that the SA node isn't firing as well. So looking at the PR interval, here let's take a look at, I believe, let's look at the junctional rhythm. So junctional rhythm, here you don't see the SA node. P waves, you don't see where the P waves should be. They should be over here, but they're actually masked. The length of the PQ interval happens to take place somewhere during the QRS segment. So now that it's not taking place, all you're really seeing is the missing P wave. So all you see is your QRS and then your T. P waves are not actually seen. Now, the second degree heart block, this basically is saying that the atrial depolarization that takes place, not all of them make their way over to the AV node. So SA node is firing 100 beats per minute. Every time it beats, it should trigger the AV node to fire, which is going to result in ventricular depolarization. Here in the second degree block, there's a block at some point. If I'm going from SA node to AV node to the ventricles, the block is taking place that's preventing the SA node firing to meet the AV node. So in the second degree block, here you'll see P, QRS, and T. Great. That's taking place normally. Everything from here to here is normal. But notice this next segment over here. There's our next P wave, and there's no R wave taking place. So there's no ventricular depolarization. This next one, from here to here, these are okay. These are not okay. So here what we see is a one-to-one. The SA node fires, QRS, and then T wave takes place. The next P wave starts, the SA node fires again, nothing happens. The next SA node fires again, and you go through a complete cardiac cycle. The next one takes place, and that's missing. So this is resulting in sort of skipping beats. In essence, what's happening is the ventricle isn't getting triggered enough because of the delay, the block between the SA node and the AV node. The last one, basically what's happening in a third-degree block, this is a complete block between the SA node and the AV node, where the SA node is firing and the AV node is firing, and it's just firing at mismatched times. So what that is going to end up doing, atrial and ventricular contractions are taking place at different times, and it results in atrial as well as ventricular muscle cells firing independently from one another. So this is where typically you would get a pacemaker installed in order to get the SA node to start firing again. Looking here, you'll see ventricular fibrillation. This is basically saying there's no pattern to the ventricles firing anymore. They're just kind of firing at random rhythm. It's a little chaotic. It's spread out all over the place. You might still get some blood flow, but it's not efficient. You're not getting efficient cardiac cycles taking place. Now, looking at blood pressure. Blood pressure we've been looking at, it's all going to be related to being able to measure pressures that are taking place from ventricular diastole, which we see the pressures in the vessels on the output of the heart, the aortic pressures. We want to measure the fluctuations in diastole, as well as what's happening during ventricular systole. So the pressures we want to measure are going to be in the aorta. And really, if I want to really be able to determine what the pressures are, that would be mimicking the information that's being sent out by our baroreceptors, would be to stick something into the aorta and measure the pressures. I would get a balloon in there and I want to say, okay, these are the high pressures and these are the low pressures, our systolic pressure and our diastolic pressures. Maybe not really the most, probably a little too invasive. So instead what we can do is measure the pressures close by. So what we can do is to, instead of measuring directly in the aorta, we can measure the pressures downstream, down an artery. And the typically place that would be the easiest would be to measure the pressures taking place in the brachial artery, since it's close to, pretty much in terms of pressure levels, close to that of the aorta. So here we can measure the pulsations, the pressures that take place. And we would do this with utilizing a sphygmomanometer, which is going to measure the pressures taking place in the heart. Again, I'll talk about that in the next, in our vessel lecture. Systolic pressure is going to measure the highest pressure, which is during ventricular systole. The diastolic pressure is the lowest pressure during ventricular diastole. And again, that represents the highest pressure for ventricular systole and the lowest pressure during ventricular diastole. So we should expect the differences in pressure from 120 down over to 80. And those pressure changes that you're seeing that take place, you should be able to see. If I were to take a pressure sensor and place that in the aorta, you should be able to see these fluctuations that take place. And each time you get those pressure waves, each of those are cardiac cycles, and that's the ejection of blood. So that ejection of blood, you should be able to see the top, the highest pressure, which is about 120, bring us back down to our lowest pressure to about 80. So looking at the blood pressure over age, like I said, 120 over 80 is ideal. But what happens with aging is that our blood pressure starts to go up a little bit higher. So about 30s, 40s, you'd see maybe about 125, 130, over 80, 85. As we get a little bit older, it gets even higher. And that we'll talk a little bit more about in our vessel lecture. Not entirely important. Recognize that females actually have lower blood pressure than men. So it wouldn't be uncommon for women to have about 110 over 70. Looking at the next slide over, you can see what the normal blood pressure is. And when we see that where we spend most of that time in, if you were to take a look at the pressures during an entire cardiac cycle, you would see that we spend about most of the time at about 80 millimeters of mercury. The other time is at about 120. So the easiest way of looking at where the average pressure is not to do 120 plus 80 and divide that by 2. That's not how you figure out your pressure, the average pressure in the aorta. The best way of measuring what that aortic pressure is to utilize this equation. Okay? So now what we're going to start doing, especially in physiology, is start to use some relatively easy calculations. What we're doing is measuring the highest pressure, our systolic pressure, minus the diastolic pressure. So 120 minus 80, which is 40, divide that by 3, which is 13.3. So 13.3 is the number that I come up with. And why do I divide it by 3? Because about 2/3 of the time we spend in diastole, about 1/3 of the time we spend in systole. So divide 120 minus 80 divided by 3 and add that to your diastolic pressure. So 80, which is our diastolic pressure, plus 13.3 comes out to about 93.3. That is your mean arterial pressure. And that mean arterial pressure is going to be important because what we're going to be looking at is that pressure is going to get conducted all the way down. We need to have that pressure form move all the way from your aorta, all the way down to your capillaries, all the way back to your veins. Because what happens if that pressure decreases, let's say down to about, this won't probably be practical, but if you bring it down to about 40, by the time this starts to dip, and we'll explain in our vessel lecture why pressures start to decrease when we go from our arteries down to our capillaries, once we get down to our arterioles or capillaries, you'll notice that the pressure will be down to 0. And if we don't have any pressure down at the capillaries, how do you expect to get fluid exchange to take place down at the capillaries? So this is why it's an important measure. The mean arterial pressure is something that they want to be able to estimate how well blood exchange is taking place down at the organs and all of our active tissues. This is going to eventually drive the gas exchange that takes place on that layer. So as we see the pressures, we see our systolic pressures. These are measurable changes that are taking place in the aorta, the arteries, and again, something that we'll start to see as it decreases by the time it makes its way down to the capillaries, venules, and veins. And again, we'll explore what's taking place in the capillaries at the tail end of our vessel lecture. We want to be cognizant of the pressures right in that area, because that's going to affect the amount of fluid that moves in and out of the capillaries, as well as the gas exchange that takes place. In the cardiac cycle, the thing that they do during the cardiac cycle, easiest way to be able to listen to the easiest way to at least derive normal function of the heart is to listen to the valves. So while we're not really looking at the pressures anymore, we want to be able to hear how the pressure, or at least how the valves are opening and closing. So what they do in a process we call auscultation is to listen to the heart. And what we're doing when we're listening to the heart is we're listening to the valves. And so if you've ever come in with anything, or you have a physical exam, one of the things they want to do, that a doctor wants to do, is just listen to your heart sounds. And the way they can do that is just place them in areas right below the second rib, to the left and right of the midline, and right by the fifth and sixth, in between the fifth and sixth rib on the left side. And that allows us to listen to the valves. So here you can see our tricuspid to the right, you can see our bicuspid to the left. We know that blood is being ejected from the atria, from the right atria down the right ventricle in this direction. So the best place to listen is not on where the valve would actually be. It's a little bit downstream of where the blood flows. If you were to take a kink in a hose somewhere, the best place to listen for any sounds in the hose is not directly where the kink is to listen downstream. It's going to be the loudest, and that's why they would measure the sounds away from the heart itself, basically where it projects. Same thing with our aortic valve. aortic valve, blood is being ejected by the ventricle up towards the top right of our body. So therefore, they're going to want to measure a little bit higher up. So all in all, what doctors are doing, or nurses, or whoever is trained in auscultation, what they want to do is they want to be able to listen to the heart sounds. One of the ways they were able to do that before, prior to the use of stethoscopes, was to actually place, a doctor would place their ear right up on the chest and listen. That became a little invasive, especially for females. So what they were able to do was utilize a stethoscope. A stethoscope basically does the same thing, but it directs the sound directly into the cup, which directs that into the ear, so you can you can hear the sounds without actually having to be that close up. So with that came the stethoscope, and what they can do is listen to the heart sounds. And the heart sounds you should be able to hear are the lub-dub, lub-dub, lub-dub. And all of that is corresponding to the sounds of the AV valves and our semilunar valves. Now going through, on the next slide over, you can see our normal ECG tracing. P, Q, R, S, T. And what we're listening to are the sounds that take place. And this is something that the Wiggers diagram will also do as well, is they'll place these heart sounds. So listening to the very first portion, you get, we're not really looking at the atria, we're looking at QRS. QRS marks the beginning of ventricular systole, ventricular depolarization. Ventricular depolarization is the start of ventricular contraction. So if I've got my, and let me just draw a simple heart, and this is a really, really simple heart. If this is going to be my AV valve, and this is going to be my semilunar valve. The first sound I should be able to hear during the very beginning of ventricular contraction. Ventricular contraction involves the slamming of the AV valve. Remember, the very beginning, prior to all this, the PQ interval represents atrial systole. Blood is being ejected out of the atria into the ventricles. So ventricular filling. Once we reach QRS, that marks the beginning of ventricular contraction. So what's happening at that point? As contraction takes place, this closes. So this is going to be our lub. So AV valve closes. The second sound, as we get blood ejecting out, and here's our aorta, blood is being ejected out into the aorta. There shouldn't be any sound in the first place. Blood is flowing normally through. Then, when the ventricle relaxes, which is marked during ventricular repolarization, is when we get the aortic valve to close, which is the sound of dub. So lub-dub, lub-dub, lub being the AV valve, dub being the semilunar valve. That should at least tell us if the valves are working normally. So that tells us a little bit about it. That hearing lub-dub lets us know if the valves are opening and closing. The other things that you would also should not hear are the sounds of turbulent flow. So take a garden hose and make sure it's completely straight, like a perfect garden hose, and put your ear up to it. What you shouldn't be able to hear, you shouldn't hear any sounds. Because smooth laminar flow just causes fluid to flow through easily. Take a kink to the hose and now listen to it, and what you're going to hear is you're going to start hearing a whooshing sound. That's the sound you're hearing. That's turbulent flow. Turbulent flow is the sound of water hitting that kink in the hose and spiraling all around in that area. That's the turbulent sounds that you're hearing. Turbulence in the heart is not what we want. We want blood to flow all the way straight through. Any sounds are bad. So what you should be hearing are lub-dub, lub-dub. Now, the turbulent flows. There's ways that we could have turbulent flows. Stenosis is the incomplete opening of a valve. So this is a valve slightly open. That's the whistling sound that you hear. Okay. Insufficiency is incomplete closure of a valve. So while a valve should close all the way, the backwash of blood from the ventricle back over to the atria has this whooshing sound. So think about what that sound would sound like. If I were to have the valve open, I have the door open, I have blood rushing out. Once the aortic pressure is highest, you should slam the door completely, and that's the dub sound you hear. Now, if this doesn't close all the way, you're going to have some blood rushing back in, right? So that is what we call our insufficiency. It's the inability for the valve to completely close, causing a backwash. So what you would hear, if this were the AV insufficiency, you would hear lub, swish, dub. That swish is blood flowing in, lub closing, and then swish, blood coming back into the atria. If you heard lub, dub, swish, that swish is taking place after the semilunar valve closes. So that is, again, incomplete closure of the valve. Stenosis is the whistling sound. So if you were to hear a whistling, so a lub, whistle, dub, and all of that is listed out for you right here. A lub, whistle, dub is going to be stenosis of the semilunar valve. So lub, whistle. So lub, that's blood squeezing through a narrow semilunar valve. If you hear lub, whistle, dub, that's blood whistling through a stenotic AV valve. Okay? So all of that is letting you know the sounds that are taking place in an abnormal auscultation. So for those of you that are pursuing a nursing degree or a medical degree or whatever degree that's involved in cardiac function, these are important things to listen for. And all of that you can get just from simply listening to the heart. Okay? I covered this. This is going to go into ECG abnormalities, which we'll skip. I'll go into a little bit about our cardiac cycle, but more so in the heart sounds, and then cardiac cycle. So what we talked about on Tuesday entirely was all about the electrical activity of the heart. And by looking at the ECG, we know that this is going to measure the rate changes in the membrane potential. And so we were able to discern from looking at the ECG that the P wave is going to be our ventricular, not aortic, atrial depolarization. QRS is going to be our ventricular depolarization. And T was going to be ventricular repolarization, which you guys already were able to answer from our quiz. So this marks the entire period. Everything from QRS marks the point at which ventricular systole takes place, or contraction. Ventricular contraction. So don't get confused that T is contraction or QRS is contraction. It's the entire period during. And so what we would expect to see in the ventricles, when we look at the action potential that takes place, QRS marks the depolarization, which we can clearly see what's happening in the purple line. QRS marks ventricular depolarization, which we see right above. We see a sharp increase in our membrane potential, in which we're going to have sodium rush in. We see the extended depolarization take place in between the QRS and the T segment. So the ST segment represents the time in between for ventricular systole or contraction to take place. Here we see the extended depolarization due to calcium influx with simultaneous potassium efflux. That's potassium leaving the cell as well as calcium entering. So that's going to keep us at about 20, 0, 10, 20 millivolts, and that's going to bring it right back down at the beginning of ventricular repolarization is when we start to see repolarization taking place. And looking at that point on, again, this representing an entire cycle from P to the next P wave, which marks, again, the very beginning of atrial depolarization. We'd also want to take a look at the pressures. So while we start ventricular depolarization at QRS, watch what happens to the pressures. The pressure in the ventricle is at about 0 prior to the contraction taking place. We see a marked increase, in pressures. We go from 0, 20, 40, 60, 80. Once we reach that point, you will see, actually before I even talk about that, you'll see when pressures continually rise. That means that as we start moving through that extended depolarization, pressure keeps rising, and that's because the ventricular muscle mass is continually contracting and contracting even more and more. And it continues all the way up to the point when we reach the end of the contraction, right up at that point. That's at our peak. And that peak pressure at that point is at about 120 millimeters of mercury pressure. That's how much force is generated by the ventricular walls during ventricular systole, especially towards the very end. Once we get into ventricular repolarization, you'll see the pressures start to decrease rapidly. And that again marks the point at which repolarization, the ventricular muscle walls relax. It continually relaxes even more up to the point that we're back to 0 millimeters of mercury back in the ventricle. Now, what does that mean in terms of the ejection of blood? We'd actually have to see, as I mentioned prior, what governs the movement of blood from the ventricles over to the aorta is all due to pressure. Okay, if that's blood moving from the atria, from the left atrium to the left ventricle, or from the left ventricle to the aorta, it's all going to be governed by pressures. Do we have enough pressures in the prior chamber to push blood out into the next chamber? Meaning, in order to get blood to eject from the left atrium to the left ventricle, the pressures have to be higher in the left atrium to push blood into the left ventricle. And then, subsequently, looking at getting blood from the left ventricle over to the aorta, we have to generate enough pressure in the left ventricle to exceed that of the aorta to get blood to move from the left ventricle over to the aorta. So do we see that happening? Yes. Right up at that point, you'll see that the back pressure in the aorta that's being exerted on the aortic valve, okay? The pressure that's being exerted on the aortic valve is saying we need to exceed that ventricular pressure above aortic pressure to get blood to eject. And when does that take place? Somewhere in the very early stage of ventricular systole, you'll see that the pressure starts to rise up to the point that we're above aortic pressure. So at that point, that characteristic point in time, we'll see the aortic valve open. And as long as ventricular pressure is above that of aortic pressure, we'll see the ejection of blood take place. But as ejection of blood is taking place, we'll see that pressure is still continually rising. Why is that? Because we're still in a depolarized state. We're still in ventricular systole. So as we're continuing systole, you'll see those bridges continually contracting. The muscular bridges continually sliding against each other, and the contraction working even harder and harder. So up at that point, you'll see when our pressures, when we reach ventricular repolarization, again, the point where that's marked on the ECG as the T wave, you'll see our pressure start to drop. And as pressure drops, we get to the point that it drops below that of aortic pressure. Now, aortic pressure is higher than that of the left ventricle. So I already mentioned that the direction of blood movement is governed by pressures. So why doesn't aortic blood move, or blood that's in the aorta, move back to the left ventricle? And that's due to the aortic valve closing. In other words, to use an analogy, if the pressure was higher in the hallway than it is in here, that door would slam shut, okay? And if that aortic valve is working like it's supposed to, that means that blood can only flow from the aorta out down to the ascending aorta, down to the descending aorta. It can only move out one way. And then as I mentioned prior to our Thursday's lecture, the aortic valve doesn't have any structures to prevent it from flipping back open and allowing blood to regurgitate. And that regurgitation of blood would mean aortic blood going back into the left ventricle. Why is that bad? Well, now you have blood that's going in the wrong direction. That's blood that should be going through the systemic circuit, but that means that's blood flowing backwards. So I'm going to take that back into, now that we've been able to establish the pressures and what the pressures mean for the ventricle over the aorta, we'll expand on this diagram by looking at the heart sounds, and then we'll also take a look at the volumes of blood in the left ventricle. So the pressure changes in the heart sounds that we would hear, in a perfectly normal heart, what you would be able to hear is basically nothing. Like I said, the heart, the valves, anything that's moving through, blood moving through a vessel shouldn't make any sounds. And if you get a garden hose that's completely laid out, completely straight, and you had water flowing through it optimally, it would create this laminar flow, meaning it flows equally. There's no, the water isn't flowing any other direction other than outwards. And that shouldn't create any noise. It's not until you actually kink that garden hose that you can actually start to hear turbulent flow. That turbulent flow is something that you don't want to hear in the heart, because now you've got blood that's not really flowing the direction it should be. It's twisting and turning, and it's not moving in that right direction. So the sounds that you typically would hear, the heart sounds are going to be important because they're going to give you the sounds that you're hearing in the valve. And the sounds that we would typically hear, you would hear the lub-dub, lub-dub, lub-dub. And all of those correspond. That first lub that you're going to hear are the slamming of the doors. Those are the slamming of the valves. And the first valve that I had described earlier, well, actually, technically before this, the valve that you would hear at this point, in the very beginning, at ventricular depolarization, is when the pressure starts to increase in the left ventricle above that of the left atrium. So that's going to slam our bicuspid valve. So this is our bicuspid valve closing. So the bicuspid valve, again, the tri-right, bi-left. Bicuspid valve closes because ventricular pressure rises. That prevents the flow of blood from the left ventricle back to the left atrium. Then the next valve that we're hearing that's going to shut is the aortic valve, hence the dub. So lub would be the first sound you hear. The second sound you hear is the dub sound. And that sound is the aortic valve closing. And then you don't hear anything for a while. Lub dub, all the way back. Lub dub, and all the way back. So that is all you should hear during direct auscultation, basically listening to the heart sounds. Now, again, this is what's taking place in a perfect heart. But there are some issues that can take place with the valves themselves that would result in turbulent flow. And the first of these would be insufficiency. That's incomplete closure of the valve. And that is what I call regurgitation. That means if that door is supposed to slam, the aortic valve is supposed to close, but it doesn't completely close. What's going to happen is you'll hear the slam, and then it opens up a little bit, and blood is going to swish back from the aorta back into the left ventricle. So what you would typically hear in a insufficient semilunar valve, and this is going to be specifically the aortic valve, you would hear a lub-dub and then a swish of blood going back into the left ventricle. If you were to hear a swish that takes place lub-swish-dub, we're going from the point at which the bicuspid valve closes, but we get that insufficiency, lub, and then a swish of blood back in, basically means blood going back to the left atrium. So just by listening to that sound, insufficient AV valve creates that regurgitation that you would hear. So again, just listening to the sounds would give you an indication as to what is malfunctioning in the heart. And again, most of the things that will malfunction really would be the valves. So that's insufficiency. Again, the door not completely closing and allowing stuff to flow right back in. It's basically backwash. That's all that's taking place. Now, stenosis, on the other hand, is an incomplete opening of the valve. It's basically a narrowing of the vessels. You ever hear of a stenotic aorta or stenotic, any stenotic valves? It's basically a narrowing. And that would be something like if you're supposed to blow out of pursed lips, that's supposed to sound fine, but it's not until you really narrow out the valve where it starts to kind of sound like a whistle. Okay? So that's exactly what's happening with stenosis, is that now that you have a whistle taking place, that's going to create some turbulence that you can hear. And naturally, stenotic valves will have a whistling. So what you would expect to hear in terms of the whistling is different. It's not like what you heard with our regurgitation. If we hear lub whistle dub, that's the whistling that's taking place after the valve closes. So bicuspid valve closes, lub whistle, the whistling that's taking place is blood moving through the next valve. And that next valve in between the left atrium, from the left ventricle onto the aorta is the aortic valve. So a lub whistle dub is the whistling of blood before the aortic valve closes. So that notes that that is a stenotic aortic valve. The other way around, which is a lub dub whistle, would be everything after the aortic valve closes. Now you've got blood moving back from the aorta, all the way going back through, eventually through the pulmonary circuit back to the left atrium. And then from the left atrium to the left ventricle is everything that's taking place is a stenosis that's taking place in the bicuspid valve. Okay, so again, narrowing of this valve creates that whistle that you hear. So lub dub whistle is everything that happens right before this point, before the bicuspid valve closes. And again, going back at this chart, lub dub whistle, stenotic AV valve, lub whistle dub is stenotic semilunar valve. So those are the sounds. And again, the sounds, looking at that, what I've been able to describe, this slide kind of gets back, and this is where we left off on Tuesday. This is pretty much just a reiteration of what we talked about. The cardiac cycle is everything from our P wave just up to the next P wave. So this is one entire cardiac cycle. And one cardiac cycle, the length of the cardiac cycle represents one beat. So you can measure how long it takes for one cardiac cycle. And if it's 0.6 seconds, that comes out to 60 divided by 0.6. It's going to be about 100 beats per minute. If it were 0.8 seconds, 0.8, that would be 60 divided by 0.8. That gives us 75 beats per minute. If this whole cycle took 1 second, how many beats per minute is that? If it took 1 second, one cardiac cycle per second, how many beats per minute is that? 60 beats per minute. If you didn't get that, that's okay, because I mentioned that even to med students, and they were like giving me blank stares. I'm like, it's 1 second for one cardiac cycle, 60 seconds per minute, 60 beats per minute. So that's okay. So keep that in mind when we look at the length of the cardiac cycle. And again, what's going to govern why, what we'll talk about later when we talk about the length of the cardiac cycle, what's going to govern the changes when we look at sympathetic output versus parasympathetic output in terms of adjusting the length of the cardiac cycle. Where would we expect to see that? And I'll talk about that in just a minute. So now that we've established the cardiac cycle, we know that systole and diastole refer to contraction and relaxation. And depending on what you're talking about, atrial systole would be atrial contraction. Okay, at least the beginning of, atrial systole is the contraction. If we're talking about ventricular systole and ventricular diastole, that's relating to contraction and relaxation of the ventricles, which ultimately is the most important part, because that's what's going to send blood out of the heart. So this slide just basically talks about what's taking place during the cardiac cycle, where you have the atrial systole, which contracts the atria, relaxation of the ventricles in order to fill the ventricles. When we get atrial relaxation, we're going to now have ventricular systole taking place. And the last portion of the cycle we'll talk about is when both chambers are relaxed. So during that contraction, again, you saw the contraction taking place in that earlier slide. Everything outside of that systolic portion, all of this, up to that point, is diastole. And ventricular diastole is important because this is the time that we take to fill the ventricles. If this is where we're starting to empty, this is where we're filling. What we talk about in this particular slide, the whole point of understanding filling and ventricular ejection, we'll get into the next stages. What's important in measuring afterwards, to give us an idea of how much blood is flowing through, is to also determine the pressures that are measured, because the pressures are going to be important, and I'll explain more in our vessel lecture. The pressures are going to be important, then we want to measure that in the aorta, as well as the pulmonary trunk. And we want to make sure that they're normal values. And so this is important. We want to make sure that aortic pressures are high enough in the aorta, because that's going to be able to push blood all the way out through the systemic circuit. And again, where I was having you guys measure this before, is mean arterial pressure. We knew that the very peak of ventricular pressure was 120, and diastolic pressure, not so much in the ventricles, but in the aorta, was about 80. So here you start to see the 120 over 80. We're just starting to touch where that 120 over 80 comes from. So the highest pressure in the aorta is 120. The lowest pressure in the aorta is 80. And that's going to be important to be able to measure the mean arterial pressure. We want that mean arterial pressure to be about 90 millimeters of mercury in order to be able to push blood. Remember what I said about the direction of blood flow is going to be governed by the pressures. And as long as pressure is higher in the sections prior, it's going to push blood all the way out through. And by the time we make it to the capillaries, we want it to be about 35 millimeters of mercury by the time we reach the very beginning of our capillaries. And that's what I hope to at least cover by the end of today, because this is where we're going to start to have the area, the crucial area for gas exchange, fluid exchange to take place in the capillaries. And and as long pressures are normal, by the time we get all the way back to the vena cavas, we can continue back again. So with those pressures in the left ventricle and as well as the aorta, we want to be able to measure this. I'm going way too far. We want to be able to measure those in what's called the cardiac catheterization. And the cardiac catheterization utilizes what's called a Swan catheter, and basically it is it's a catheter. It's a tube with a balloon at the tip, and that allows us to basically tell pressures. It's a form of a baroreceptor. Okay, and that balloon that gets fed in, it's actually created by a guy named Dr. Swan, who's actually at UCLA. I believe he may have passed away. I don't know if he's still alive, but I know he's old. But nonetheless, we want to be able to measure the pressures directly from the aorta. We can typically do that. Again, what our lab is going to be talking about when we do it next Thursday, the lab is going to have us measuring those blood pressures in a cuff that's close to the same level as the aorta. It'll give us an indirect measure of what the pressures are in the aorta. But if we want to measure it, we actually would directly measure it by feeding this Swan catheter. We would cut the femoral artery, we would place the tube in, we'd feed it all the way up the descending, up to the ascending aorta, and then we can measure the pressures in there directly. And that's going to give us the mean arterial pressure, a direct measure in the aorta itself. That's something that's done, but that gives us the systemic circuit. The only way to actually measure the pulmonary circuit is to measure the mean arterial pressure in the pulmonary trunk. So this is going to be important. We want to make sure that normal pressures take place over here, because by the time that blood makes it over to the lung, we want to make sure that the pressures are normal in order to facilitate gas exchange taking place. If it's not normal, then we get a form of edema around the lungs. And that edema or that pulmonary hypertension is not a good thing, because then we don't get optimal gas exchange. So the way we can measure that is what's called a right heart cath. And a right heart cath is where they take the same Swan catheter, they make a cut right there in the jugular. This is actually an outpatient procedure, and you're awake for this. They make a cut, and they feed the catheter in, and it goes down the superior vena cava right into the left atrium. The right atrium. It's going to move right through our valve, our AV valve, and they've got to rotate this thing. And if you ever watch interventional radiologists, this is what they do. They will feed the catheter in, and they can just take a look at an ultrasound. They can twist it the right way, and they'll get that tube to curve right back up the right ventricle, right through the pulmonary valve, into the pulmonary trunk. And that gives them the ability to measure directly what the pressures are. What's interesting when they do these right heart caths is that they have to learn how to do this move. So you see them do this quick twist, but when you see people learning the first time, people are awake during this, and sometimes they end up jabbing the right ventricle, and you'll see the arrhythmia that takes place. And you'll see when you're looking at ECG, all this arrhythmia is taking place because the ventricle is not squeezing the way it's supposed to, and then the patient feels it, and they're like, yeah, you're kind of stabbing my heart a little bit. Can you move it the right direction? And then they get it. So that's the whole thing of why we want to measure these directly, in order to tell us what the pressures are. While aortic pressures, a left heart cath we can get direct pressures. We can also do that from normal blood pressure, which gives us an inference. We can't do it for the pulmonary artery. This is their gold standard for being able to diagnose somebody with pulmonary hypertension. Let's take a look directly at the pressures in the pulmonary artery. All right, so now that we've described the cardiac cycle, what we're going to start to do is look at the events that lead to, because I want to be able to pair this up with the ECG. I want you guys to be also well aware of where the sounds are taking place, and then most importantly, how the filling and emptying takes place in each of the chambers. Again, our primary focus is really on the ventricles, so we'll be looking at the left ventricle filling and emptying as a point of reference. So like I said, the first section, we can take a look at what's taking place by defining systole and diastole. So looking at the first section, I'm going to draw atrial up here and ventricular down over here. So atrial, this is going to be systole. Again, systole is marked by contraction. So this is the very beginning of the P wave up to the QRS. And at this point, pressures are higher. I'm just going to draw an up arrow noting where the pressures are the highest. And the pressure being high during atrial systole will force blood out into the ventricles. So this is the portion where ventricular filling is taking place. So at this point, this is going to be ventricular diastole. This portion right here below marks ventricular systole. And how do we know that? Well, where we can see isovolumetric contraction. So this starts the very beginning at QRS, where ventricular depolarization takes place, and therefore pressures are starting to rise in the ventricles. And you'll see that there's these two different areas that I'll describe in just a little bit. So ventricular systole, the rest is going to be ventricular diastole. If it's not contracting, well, if it's not contracting, then it's relaxing. That's all that's taking place. So this marks systole. This is the marked area. Now atrial systole, the only portions of atrial systole, all of this is atrial diastole. All the way back out. Up to that point. And this is back to systole. So, looking at this is again what I'm going to describe are the chambers. Atria being my left hand, ventricles being my right hand. This begins atrial systole. P wave takes place, SA node fires, atrials start to contract. That's going to squeeze blood into our ventricles. So this is where ventricular filling takes place. Now, once we've filled it up, we're going to now move on to the QRS segment, where QRS is going to be atrial repolarization and ventricular depolarization. And during ventricular depolarization, which is ventricular systole, at least from systole, in between ventricular depolarization and repolarization, when systole takes place, I'm now squeezing the ventricles. But what I'm doing is I'm raising the blood pressure enough to first close my AV valves, and I'm going to continually generate pressure in order to exceed that of the aorta. So the aorta we saw about 80 millimeters of mercury. That's the back pressure on the aorta. 80 millimeters of mercury. So once I exceed 80 millimeters of mercury, now I'm getting blood ejected from the left ventricle into the aorta. So hence why you're going to see what's called isovolumetric contraction, and then ventricular ejection. That's the ejection of blood that's taking place at the latter half of ventricular systole. So now that I'm squeezing the ventricles, we're now going to get to the T wave where we get ventricular relaxation. And now we're going to go through a period of isovolumetric relaxation. That means no volume is changing. No volume is changing in the ventricles. Now the muscles are relaxing, pressure is decreasing rapidly, and then we go through ventricular filling. And then we continue with ventricular atrial system. So if I play that back, squeeze, squeeze. Okay, see how that works? All right, so now that we have that down, where do we expect to see the sounds that we would hear? Lub-dub, lub-dub. The first one, as I mentioned earlier, the very beginning of isovolumetric contraction is when we see pressure is starting to rise in the ventricles, and that's going to slam our AV valve shut. So we expect to hear the first sound of lub right at that point. Ventricular ejection is blood moving out from the left ventricle out to the aorta, and right to the point when we get to the T wave. T wave takes place at this point, and then ventricular muscles relax. They start to relax, and therefore the pressure in the aorta is higher than that of the left ventricle, slamming the aortic valve shut, causing that next sound, the dub sound. So lub, dub. And then we're going to go continue on with isovolumetric relaxation, ventricular filling. Ventricular filling naturally taking place because now you've got blood filling up in the atria, and now blood is going to flow from the atria over to the ventricles naturally, and then we get that last bit where the atrial contraction comes in and squeezes the last bit of blood in the atria to fill the ventricles. So now that we have the system in place, I've talked you through the ECG, I've written out where the sounds take place. What we'll do next, and here you can just do it off the ECG itself, and I can do that one more time. These basically point out when the valves are opened and closed, just as I described earlier. So you would want to naturally pair these up by looking at the sequence of events. We would see that in this slide where ventricular diastole takes place, the ventricular diastole, and atrial systole marks that first section, and I'm referring to this slide over here. So the AV is open, our AV valve is open, and our semilunar valves are closed, which we mark over here. Ventricular systole, during this section right here, you'll see that our AV valves are closed, while our semilunar valves are open. And that's really taking place right at the very end, okay? Towards the latter half of ventricular systole. AV valves are closed, but the valves are open to allow blood to flow from the left ventricle to the aorta, or the right ventricle over to the pulmonary trunk. And the last portion of that section right here, ventricular diastole, is also taking place at atrial diastole as well. And here we can see the valves being open. So if this is diastole, this is atrial diastole, and this is also ventricular diastole. Here you can see the AV valve open and our semilunar valves closed. So that names it all off, and you can use this to dictate what's taking place. Here again, this breaks down the different sections, so this should be paired up the way we described earlier. We can see where ventricular systole is taking place. You can see where ventricular diastole. And as I mentioned earlier, this is going to be the point at which ventricular contraction from here to this point. One thing I want to mention also is this period in between the T wave up to the next P wave also takes place. All that is diastole for both the atria as well as the ventricles. So what's happening at this point in time? This is blood filling up the heart. This is blood as it moves through. We've ejected it during ventricular systole. Now imagine that pathway of blood as it moves from the ventricular systole. You've pushed blood out to the aorta, and that blood is making its way through all the way from the aorta down to the arteries, to the arterioles, to the capillaries, to the veins, back to the heart. All of that is coming, pushing blood down that entire pathway to fill it back up. So we've got blood returning back to the heart. So if I can just draw out the heart, again, I know it doesn't look like that. I mean, if you can picture what it's supposed to do is, if I'm squeezing the heart at this point and pushing blood out, think of all that blood, just like if I can think of be a good analogy for this, like a little train set. If I'm pushing fresh blood out, that's going to continually push blood all the way back in to return back to the heart. So while I start ventricular contraction, that's starting to fill back our atria. And as we're filling up the atria, once we get to the point at the end of ventricular systole, ventricular diastole takes place, and now we're going to start to continually fill. So during this point, the length of the cardiac cycle, everything from the P wave all the way out to the next cycle, for the most part, is always going to remain the same length. Well, let me refine that. Everything from the P wave to the T wave is going to remain, for the most part, the same length. We want to make sure that still remains the same length because we want to give it enough time for atrial systole to take place and ventricular systole to take place. If I try and shorten this too much, specifically ventricular systole, that means I'm not going to have enough time to eject all the blood that I need to. So we want to make sure that that time remains the same. And during times at which we start to see increased sympathetic output, we would still monitor the ST times. The ST time still indicates that we have enough time for ventricular systole. We want to make sure we want to get as much blood out of the ventricles. So now that I've kind of talked about the sympathetic effect, how do we speed this up? If this is over, if the length of the cardiac cycle from P all the way to the next P wave is about 0.8 seconds, which comes out to 75 beats per minute, what happens if heart rate increases? What happens? What does the sympathetic effect do? How do we decrease the length of this? Well, the time in between the T all the way over to the next segment, the next P wave, is the time at which the heart is at rest. It's waiting for the next depolarization to take place. I've just repolarized our ventricles. That entire conduction pathway has started from the SA node all the way out to the Purkinje fibers. Now we're waiting up to the next point when we get the next trigger for atrial repolarization to take place. So during that time, when we start to see the effect of the sympathetic pathways, it would actually cut the amount of time in between the T wave to the next P wave. I'm going to shorten the time during this point in time. So you might think, if I'm going to shorten that amount of time, doesn't that affect how much blood is filled up during this period? And we'll talk about that more towards the end of today's lecture on how we can compensate for that lack of time. So in other words, to sum up this slide, the length from P to T is almost always going to stay the same. In between T and P, between while we're in atrial diastole and ventricular diastole, is that sort of rest time that we can cut into. And if I can cut some of this time off, that just gives us enough time to have the next beat. And I'm going to start to decrease the length of our cardiac cycle. Or in other words, decrease the time in between P waves to the next P wave. And that effectively will speed up our heart rate. So sympathetic pathways tap into that. What questions do we have about that? Yes?
So from the T to the P, once is the heart arrest, and once it reaches the P again, so that was atrial repolarization?
Yeah. I'm sorry, atrial depolarization is the start of this, and we start the whole cycle all over again.
Okay.
So again, the P wave begins when atrial contraction takes place. When atrial contraction starts, which is atrial depolarization, that's when the SA node fires again. So we want to cut time in between. Okay. Any other questions? So on to Wigger's diagram, tying in everything that we've just talked about with atrial systole, ventricular systole, ventricular diastole. I want to talk about these ventricular systole and ventricular diastole. And you can note that these segments are noted by the purple segments. And if we take this diagram and draw it all the way back up, this will list out all the cardiac events that take place. The cardiac events in related to the electrical activity of the heart, the ECG. Looking at the electrical activity that, or at least the pressures that are taking place in the left atrium, which is marked in green. Our left ventricular pressure is marked in blue, and our aortic pressure is marked in red. So this is electrical activity, this is our pressure. These are the heart sounds. And again, heart sounds generated by the sounds of those valves slamming shut. And then we want to tie that in to the volume of blood in the left ventricle. So all of this is going to be described for you on these slides over. Cardiac cycle, all the way down to the very end. We'll kind of describe that for you. So like I said, I will walk you through it, but if you need later reference, this is what you guys can read through. So the very beginning of all of this, we can mark when our bicuspid valve closes. That's going to be our first lub. The second sound right here at S2, which marks the end of ventricular systole, is when our aortic valve closes. Hence, we have our lub dub. So we can work off those sounds, which we've already been able to describe. We've already measured the pressures. We've talked about the pressure in between the very beginning of ventricular systole and the very end of ventricular systole. We've seen the pressure changes that take place in the left ventricle marked off by blue, and how that relates to the pressures in red, which mark our aortic pressure. In order to get blood to eject, note this very beginning segment. This is the very beginning of ventricular systole. This is called isovolumetric contraction. And it is called by isovolumetric contraction because this is the same volume. Same volume. There's a marked increase in pressure in the left ventricle. But why the volume isn't changing is because our aortic valve remains closed during that time. And at the same time, our AV valve is closed. So while I'm contracting the ventricular muscle mass, I'm just taking a water balloon and I'm squeezing it and I'm increasing the pressure. And it's not until I can get that pressure higher than that of the air pressure that I can finally get blood or water to squeeze out of that water balloon. So during this time, I see a marked increase in pressure, and it's not until I get to the point that we exceed that of the aortic pressure. So remember, we go from left atrium to left ventricle to the aorta. And we know that at rest, it's about 80 millimeters of mercury. And we can see that right up here, 80 millimeters of mercury. The pressure starts to increase, up to the point that the ventricular pressure exceeds that of the aorta. Once we reach that point, we see, we're right down at this point right here, which marks the very beginning of ventricular ejection. So the only time blood is flowing out of the ventricles is when we open up the aortic valve all the way up to the point that ventricular repolarization takes place, where our ventricular muscle walls relax, pressure starts to decrease, and the aortic pressure is once again higher than that of the ventricles. And how do I know that? Well, take a look at the volume in the ventricle. Here we are at the highest We see ventricular ejection taking place. We see blood ejecting all the way back out. And we don't completely empty out the ventricles. That's not entirely possible, but we get down to the very end. So marking off that point where we see the highest amount of blood, again, the highest amount of blood would be at the end of atrial systole or at the end of atrial contraction. We now have the isovolumetric contraction where the volume remains the same. This point right here, at this point at the very end, which is the end of diastole, the end of ventricular diastole, which goes all the way, and let me just draw V diastole. We go from here all the way out to this point right here. This marks ventricular systole. So in between ventricular systole and diastole right here, this is the end volume at the end of diastole, also known as the end diastolic volume of the left ventricle. That's how much blood we have in the left ventricle right before we begin systole. So end diastolic volume. Now at the very end of systole, at the very end of ventricular systole, marks how much blood we've actually ejected out of the ventricle, which we call our end systolic volume. So in other words, the amount of blood that is pumped out of the ventricles during ventricular systole is the end diastolic volume, which is about, in this area right here, about 130 milliliters in the ventricle, all the way down to the very end, which is about 60 milliliters left over in the ventricle. And that is our stroke volume. So stroke volume, which I'll describe in our later slide, is how much blood is pumped out per heartbeat. Like I said, I'll describe that for you on our later slides. Right there, stroke volume. And it's our end diastolic volume minus what's left over, which is our end systolic volume. So that, I believe, is our second equation. The first equation being our mean arterial pressure, this being our second equation, which is honestly a really easy equation. You just have to know which is which. So given that, now that I have two equations, we've got actually a couple more, and then we're going to have more in respiratory. So this exam is going to require you guys to have a calculator. And what I'm going to have you guys do when you guys do your exam number 3, exam number 3, bring in a basic calculator. No TI 82, 3, or 5 or whatever. Just a basic calculator. You don't need to do any. You don't need to do any scientific calculations. It literally is just a times, multiply, subtract. No phones. Just a simple, easy calculator. All right. And I'll remind you guys before the test. So now that we've measured the amount of blood that's pumped out at the very end of diastole and the very end of systole, that's our stroke volume. And then once we get down to that point, right at the very end, we're at the beginning of ventricular diastole. You'll see the pressures come back down. Our first part is going to be called isovolumetric relaxation. This is where we have our AV valves closed and we have our semilunar valves closed. This is when we just see that the muscles start to relax. So we've got the same volume, which we can note on here. The volume doesn't change. Same volume, but now the muscles are relaxed. And now the pressure is rapidly decreasing. And not until the left atrial pressure is higher than that of the left ventricle do we see the bicuspid valve open right back up, and we expect ventricular filling to take place. Now you see the valves opening, the pressures are higher over here. And now blood moves from the atria down to the ventricles. Now keep in mind, at this point, we're still in atrial diastole. The atria is not contracting until we reach the next P wave. So while that's taking place, now the pressures are even higher again in the left atrium. Left atrial pressure again starts to rise up, and now we get more blood filling up the left ventricle, thus repeating the entire cycle. All of this right here just basically shows you the same thing. I posted this back up just so that you can see it more in a linear form. And here we remove the cardiac cycle, so you can at least see what's taking place during ventricular systole, ventricular diastole, ventricular systole again. Again, if you were to measure each of the different cycles, you'll see that we have 1, 2, 3, 4, 5, 6, and there's actually 7 phases. You'll see 6, you'll see 7, it's all basically the same thing. Atrial contraction taking place, you'll see ventricular systole taking place, and again, the same cycle that I showed you earlier. Isovolumetric contraction, rapid ejection and reduced ejection. This is ventricular ejection. And all this comes down to some text sources break down how fast blood is leaving versus how much is leaving. And that you can sort of measure, again, not something you really need to know. But the high pressure that takes place over here, this is the rate at which blood is being ejected. And this is the second phase where less blood is ejected at a slower rate. Not entirely necessary for you guys to know. The ventricular diastole, you'll see isovolumetric relaxation, and again, ventricular filling. And that one we can measure our slope up here versus our slope over here. And again, not really important, but when you guys get into more advanced cardiovascular system, you'll be looking at the differences in there. And the mechanics are going to be slightly different. But nonetheless, same concept. Either you're ejecting blood or you're filling blood. Takes place during systole, as well as ventricular filling taking place during ventricular diastole. So all of that is narrowed out for you. And then each of these right here will point out at the points at which the valves close. So you can start to measure where these different sounds come from. And one thing I want to point out, if you ever get curious and wonder why there's a third heart sound, that third heart sound is abnormal in people over 40 years old. But is actually common in younger people and actually even common in athletes. And that sound is the reverberation going on. It's basically an echo of the ventricles filling. That's all it comes down to. You have a strong enough heart, that blood flows right back in and it creates this echo. That's all it is. So normally you see here a lub-dub, and this third sound is a very, very minor sound. This is going to answer the question about the relation between pressure and volume. Now, this really just looks at the changes in pressures as well as the volume that takes place. And I don't want you guys to think that that they're both tied in together. If pressure rises, volume decreases or anything like that. This illustrates the separation that occurs between the changes in pressure as well as the changes in volume. So what we're looking at on our y-axis is the pressures in the pressure in the left ventricle, and what's happening in the x-axis are the pressure of the volume changes. So right off the bat, we can easily isolate when our isovolumetric filling, isovolumetric relaxation, and filling, isovolumetric contraction, and relaxation take place. And where do we see that? The areas at which ventricular volume remains the same. So over here we're about 120 milliliters, 130 milliliters to be safe. You'll see this area right here, the volume doesn't change. But we see the pressure rising. B is end diastolic volume. This is the point at which our ventricle is filled up at about 120, 130 milliliters per minute. I know that at this point the mitral valve closes, or bicuspid valve closes. So let's take a look at where that takes place. Number 2. So this point right here is where our bicuspid valve closes, and you see that the pressure is rising, but the volume doesn't change. That's exactly what's describing when we go from B to C. Pressure is rising because we see this is where ventricular contraction starts. We see pressure rising. And it's not until we exceed the aortic pressure that the aortic valve opens. And so now that our pressure is higher, we're going to continue ventricular systole all the way to the point. Now we're seeing a decrease in volume. All this taking place. So let me draw what's taking place from D to B. All of this marks ventricular systole, while this describes ventricular diastole. So ventricular systole taking place over here. We see pressure rising, but no changes in the pressure. And then the latter half of systole where the valve opens, and we see the volume of the ventricles decreasing. That means blood is being ejected out of the left ventricle into the aorta up to the point we get to ventricular diastole. So right here marks the T wave. Our valve closes, and then our ventricle muscle walls relax. And it continually relaxes. There's no change in the volume of blood. Here we are at the end systolic volume. And then, once we reach this end, this is where the mitral valve opens up, and then we're going to continue. Now we're going to continue filling. We're going to have the atrial contraction taking place and filling up of the ventricles while the pressure remains low. So all of this really points out all that's taking place. Okay. Again, each of these cycles, each of these points right here talk about the different segments. So just in case you're lost at any point, feel free to refer to that. I can also talk to you guys afterwards if you need to revisit this. What questions do we have before we continue? This Wigger's diagram and everything else, when you look at it from the perspective of the pressure and volume, is the pinnacle of it all. So if you have a good understanding of where you are now, good. If you don't, that's okay, because you got to just go over this. You got to dissect it and see each of the segments and look at it from our pressures, our volumes, the electrical sounds, as well as the heart sounds. Now piece it all together, and that will give you the entire cardiac cycle. No questions? The last thing we're going to take a look at before we go on break is to, now that we've been able to describe all of the heartbeat stuff, last thing I'll finish off with are the different time points. So here I'm just going to draw again our P wave, QRS, to T wave. P, QRS, to T. And the time in between, when we look at the length of a cardiac cycle, we're just going to use an example of 0.8 seconds. This entire length is 0.8 seconds for the length of a cardiac cycle. And if you were to break that down, the time that's required for the atrial contraction to take place, again, atrial systole takes place between P and QRS. That takes about 0.1 seconds. Ventricular systole, which takes place between QRS and T, is about 0.3 seconds. Everything else between is the remaining 0.4 seconds. And that's when all of our chambers are in systole. I'm sorry, in diastole. So they're relaxing, and everything's filling. Blood is making its all the way back and filling up that point out. So when we see the sympathetic effect is going to decrease the amount of time during this point. So this is where our fudge numbers come from. We can start to kind of decrease the amount of time in between the T to the next P wave, that 0.4 seconds. Now when you start to get into much more advanced text, you'll start to see that there are some changes that take place between PQRS and ST, minor changes there. This tells us the electrical activity. Even though there's minor changes, there's also changes that are taking place in the amount of filling. But right now, that point is moot. We're just going to stick with 0.4 seconds in between that we can allow to change. Heartbeat hypothesis, I don't even want to really talk about this. It's theoretical. I don't really like the theoretical stuff. but basically saying the size of the organism, when we go from mouse all the way out to a whale, determines the number of heart, depends on the metabolism. So we start to see an increase in metabolism, a decrease in metabolism. It's not even related to metabolism. The larger the organism, the longer the life expectancy. This all boils down to the theory of 1 billion heartbeats. And I've never really liked this, but that each organism has 1 billion heartbeats. So each organism, so-called, has 1 billion heartbeats in a lifetime, and the smaller the organism, the higher the metabolism, therefore the higher the heart rate they have. The higher the heart rate, the less length of time they're expected to live. So the more heartbeats you have, the faster your heart beats, the faster you die. And so if you look at a mouse, it doesn't live very long, less than like 2 years or something like that. You look at a lion and a whale, and they live like 60 years. Human is kind of off the charts, and of course there's so many different factors as to why humans live longer. I don't even know where turtles come in the mix. I know turtles, and I'm really just basing that off Finding Nemo. I'm not really a biologist, but I know they can live a really long time. So again, just a whole theory that I'm not even really fond on. So you know what? Scratch that altogether. If you're ever curious about it, it's called the heartbeat hypothesis. So with all that, everything that we've described, it describes everything related to the heartbeat. The electrical activity that takes place in the cardiac cycle. We know now how the heartbeat takes place and what governs the cardiac cycle. We did not talk about, especially at the very end of Wigger's diagram, we're able to take a look at how much blood is ejected in a heartbeat. Okay? So that defines our stroke volume. Not only is it the number of times our heart beats, but it's how much blood is ejected every time we beat. That gives us, and again, looking at the stroke volume is how much blood at the end of diastole, and the remaining blood at the end of ventricular systole gives us the stroke volume. Okay? So another example would be rowing, for instance. You get somebody who rows, and you'll probably see, has anybody gone kayaking before? Yeah, so if you do kayaking and you kind of row quickly, but you're barely scraping the surface of the water, you're not really moving as fast. If you move really slow and you're just dragging a bunch of water with you, sure, you've got a slower cadence, but you're moving more water with each stroke. So that's your stroke volume. What you want to picture to be optimal kayaking, and I'm hoping this analogy is working because it's the first time I ever used it. But with kayaking, you want to make sure that you get the optimal number of strokes and the amount of water that you're pushing at the same time. That amount that you're pushing is called your cardiac output. And your cardiac output is the number of times your heart beats times the amount of blood pushed with every beat. Okay? So looking at that equation, cardiac output equals heart rate times stroke volume. That is how much blood is flowing through the heart per minute. And how do we derive that? A heart rate is in beats per minute. And then stroke volume is in milliliters of blood per beat. And if you remember elementary school, whatever this is, multiplication, you cross out the beats, and that's the number of milliliters of blood per minute. So if we were to take the example of 70 milliliters per beat times 75 beats per minute, that comes out to 5,250 milliliters of blood per minute. And you just convert that back down to liters of blood, 5.25 liters of blood per minute. And that should, and what governs our cardiac output? It's basically how much oxygen you need to deliver to the active tissues. So if you're sitting here at rest and you're not using much, you only need so much oxygen. Therefore, your heart rate, we can not only our heart rate, but our stroke volume, we can autonomically regulate to match what your metabolic demands are. If you start to generate more CO2 and use more oxygen, let's say you start walking or you start running towards your car or something like that, naturally your cells are using more oxygen and you're putting out more CO2. So therefore, you're going to need to increase your cardiac output to supply those demands of O2 and CO2. So with that, when we look at that, at rest, again, cardiac output at rest is about 5.25 liters per minute. I did mention earlier in our blood lecture that we have about 5 liters, about 9 pints of blood, 5 liters of blood in a male of average height and average weight. So that means that amount of blood is being circulated per minute. If you were to follow a red blood cell and make it pass through the systemic circuit and the pulmonary circuit, it would take about a whole minute for the journey. And in your notes it says it takes about 30 seconds to make its way through the systemic circuit, but then it needs to make its way through the pulmonary circuit as well. That's a full minute. So that's a whole ride that a red blood cell becomes oxygenated and deoxygenated. And that will that kind of serve as the sort of template that we'll talk about at the latter end of our blood vessel lecture. So we can adjust these. We've talked about autonomic adjustments of the heart rate, but we need to look at the autonomic adjustments of stroke volume. And I'm going to go back to an even earlier slide that I'd like to refer to. This one's going to be important, and this one is going to be the autonomic regulation of the heart. This is slide 50. So just count 50 slides from the very beginning and you'll get to this slide. So this slide points out, if you remember on Tuesday, we talked about the pathway from the parasympathetic output. And remember, if we cut the nerve, the vagus nerve, that's the one that leads from our cardioinhibitory of our medulla oblongata, that innervates the SA node and the AV node. These are going to affect the intrinsic firing rate of the SA node and the AV node. That's parasympathetic. But sympathetic output, which travels again from the lateral gray horn out to the sympathetic trunk ganglion, or postganglion sympathetic neurons travel from the sympathetic trunk ganglion to the SA node, the AV node, and then lastly, the ventricular muscle mass. So if I have epinephrine that's being sprinkled right onto the ventricular muscle walls, the ventricular muscle walls have adrenergic receptors, meaning they receive epinephrine and norepinephrine. And that causes the muscle to contract even harder. And we want that. Imagine if you have that same water balloon and you can get it to, and you can squeeze that water balloon even harder, you're going to eject more in that heartbeat. Okay? So now going back to where we first started. And again, this is something that you want to do to refer to what's happening, because now what we're able to fill, and that's 2 slides over, the sympathetic effect on ventricular muscle is to increase contractility and strengthen the contraction. And it does so by looking at the cardiac muscle cells. We want to strengthen that. And let's take a look at how that takes place. Slides. Okay. So this slide talks about, before we get to that notion, this is going to talk about the cardiac output. Cardiac output is our at rest, the stroke volume, which you calculated about to be about 70 mils per beat, and then the heart rate. But we want to be able to measure what that maximum is. What's your maximum heart rate, and what's your maximum stroke volume? And we know the maximum heart rate is about, you guys know how to calculate your own max heart rate? I'll show you. 220 minus your age. So if you're 30 years old, your max heart rate roughly is about 190 beats per minute. It's a rough estimate. It's the lesser of our most accurate predictors for maximum heart rate. There's even a better equation, but I won't share that with you guys because it's a lot more. But 220 minus age is your way of being able to measure what your maximum is. And so take somebody who's about 40 years old. This is what their heart rate is at. Max heart rate is 180. And then your maximum stroke volume isn't going to exceed that of your end diastolic volume. So it'll be about 120, 130 mils per minute. So you times those together, and that's what your maximum cardiac output is. Right at this point is your maximum cardiac output. This is you at rest. And when you need to adjust for the amount of sympathetic activity that you're doing, you can do increases in both your heart rate as well as increases in your stroke volume to maximally increase your cardiac output. So when you draw the line that reaches out all the way to this point, this is how we can adjust both stroke volume and heart rate to accommodate increased metabolic load or increased physiological need or increased blood flow out of the heart in order to supply blood, nutrients, oxygen, gas exchange to take place, and everything else. So that's the effect of sympathetic, the sympathetic effect is to increase cardiac output by increasing both stroke volume and heart rate. Now, how does this work? So the principles that I'm going to talk about, things that are going to affect stroke volume, and this is going to be described for you on the next slide over. How do I get the heart to pump more blood? And I'm going to use the example of a water balloon again. I'm going to take a flimsy water balloon. It doesn't have to be flimsy, just be a regular one. And if I want to get the most blood to pump out that one time I squeeze that water balloon, there's two ways to do it. Number one, it's how much water I fill in the balloon, right? That's number one. How much water I fill up in the balloon. And number two, how much resistance, or even a better term, is how wide is the nozzle on that water balloon. If I have a tiny nozzle at the very tip, and I'm trying to get as much water out of that, I'm just going to have this water balloon that's going to spew a little stream of water, and that's not what I want. I want to make sure that the nozzle is big enough to allow water to flow out. So now I've got a water balloon that I'm filling up, and then I'm looking at that. That's going to describe what's taking place in our preload and afterload.
So this is describing how much what, sorry?
How much water is filling up. Think about it this way. If I take a water balloon and I fill it up about halfway, and then I let go of that water balloon, do you think it's going to force water out as hard as if I really fill up that water balloon like almost 100% of its size? If I were to have a filled water balloon versus a half-filled water balloon, which is going to shoot water out faster? The full balloon. That means the more I stretch out, it doesn't even have to be water, it could be a balloon. If I fill up a balloon halfway and let it squeeze out, it'll force air out fine, but not as fast as if I were to fill that water balloon out. This whole concept is what we call preload. The more you fill up that balloon, right? The more that you fill it out, the more it'll stretch out. And the more that you stretch out is going to cause more ejection, or therefore increased stroke volume. So increased preload means increased stroke volume. So think of it as a spring. The more I compress a spring, the more potential energy it has. Same thing with the heart. The more I fill it up, to a certain degree, I don't want to overfill the heart, but if I fill it up, it's like a spring. The more I fill it up, the more potential energy it has. And once it switches over from potential energy to kinetic energy, that's more blood being ejected out in that beat. Now, the other concept is what's called the afterload. And the afterload will make more sense when we get into our vessel lecture. This is the systemic vascular resistance. This is the resistance that's posed on number 2. How optimal are my vessels? Are my vessels narrowed out where it makes it harder to squeeze blood out? Or are my vessels even bigger to allow blood to flow out optimally? So take that same balloon, widen up the nozzle for it, and fill it up to maximum, and let it go out. And that's going to be able to eject water or air, whatever medium you're looking at, more efficiently than that one if the nozzle was closed up. And that's the afterload. And this is all tied into what's called vascular resistance. How efficient are your vessels that are delivering blood out? So now imagine, take that same concept and think of aortic stenosis. If I take the aortic valve right there and I make it stenotic, means that the left ventricle has a harder time squeezing and it's whistling that blood out of there. If blood is trying to squeeze out through that stenotic valve, that means that the left ventricle has to work harder to push blood out into the aorta. And that's going to be excess effort. And if your heart is constantly working and it needs to ramp up, that resistance is going to affect it. And so what we'll talk about in our vessel lecture is our ways that our body will reduce resistance in order to allow for optimal blood flow increase. These are things that we need to understand. And preload is something that also involves vessels. How do we get more blood to return back to the heart? So looking at all of these, again, these are the things that will affect stroke volume. Ventricular filling before contraction is a preload. How much blood can I fill? So increased preload increases stroke volume. Increased afterload decreases the stroke volume. The resistance in the blood vessels the heart is pumping into is the afterload. And then lastly, the contractility of the ventricles. Basically, how well does the ventricle squeeze?
So what would be an example of a resistance?
Resistance would be the aortic stenosis. Another example would be high blood pressure, high blood pressure is great. That's another good one. If high blood pressure, if the aortic pressure is, let's just be, for our diastolic pressure would be, I don't know, 100, and again, this is kind of 100 millimeters of mercury. That means that the left ventricle has to go higher, has to contract higher than 100 millimeters of mercury to allow blood to eject into the aorta. That means that's less volume that goes into the aorta that makes its way through the systemic circuit. And in order to get more blood out, the heart has to pump even harder and has to work more heart beats.
You have to put in more effort, basically.
Thank you. Exactly. So taking somebody who is athletic, right? And the reason they have a lower heart rate, again, cardiac output equals heart rate times stroke volume. If they've got a good stroke volume, that means their heart rate can decrease to keep up the same cardiac output. Somebody who is athletic has an optimal stroke volume. They know how to pump blood efficiently per beat. Therefore, they can bring their heart rate back down. But they can still get to their maximum heart rate. That means their cardiac output is even higher than what it normally is for a normal person. Hence why they call it cardiovascular exercise. You want to work on the heart itself. This slide talks about the effect of the preload. All of this is related to what's called Starling's law of the heart. That concept that I talked about earlier, the more the heart muscle is stretched, the more forcefully the heart will contract, just like that spring analogy that I used. So you want to have an optimal length, and looking at the sarcomere length, this is the cross bridges. You want to basically be able to pull the spring optimally so that you can contract even harder. And that would, looking at the normal heart, if we increase the preload, stroke volume doesn't really change as much in a normal heart during rest. But if we look at what's happening during exercise, you'll see that stroke volume increases quite a bit. So that increase in stroke volume due to the increased sympathetic tone is really due to the epinephrine. Same thing that we've always mentioned, epinephrine and norepinephrine. Catecholamines flowing through the system will now increase the contractility of the heart, therefore increasing the stroke volume, increasing the preload. The harder you pump, the more blood flows through back into the heart to fill it back in the first place. So the more the preload, the more the stroke volume. That's Starling's law. Stretch it out more, you'll get more out. And the more blood you get out per beat increases your cardiac output. So all of this looks into the components for our cardiac output. To take a look at the heart rate, the two different components, heart rate and stroke volume. And each of those, increased preload would increase your stroke volume. Increased contractility due to our epinephrine and norepinephrine our thyroid hormones, which are going to also play an effect on contractility. Looking at decreased afterload, you want to make sure that there's less resistance in the blood vessels, and all of that will contribute to more blood beat per minute. Now, increased heart rate, same thing. Increased sympathetic output onto the heart will drive an increased firing rate of the SA node. Decrease your parasympathetic output will also increase your heart rate. Catecholamines, your epinephrine, norepinephrine, your thyroid hormones, and as well as the other factors that come into play that would increase your heart rate. Some genetic factors play a role, your age plays a role, your physical activity, and your metabolism play a role on what your heart rate is. So combine these factors and that will increase your cardiac output. The last concept here talks about the physiological mechanism to how we get increased contractility. So epinephrine, our catecholamines that are flying around, have these receptors. These are adrenergic receptors that receive norepinephrine. And what this is going to lead to is increased contraction of the muscle cells. So epinephrine binds to our receptors. Our receptors will turn on that G protein switch, which will catalyze a reaction for adenylate cyclase to convert ATP into cyclic AMP. This whole process you've seen before for the cyclic AMP that was our intracellular messenger. That cyclic AMP comes in and turns on a protein kinase A that activates our calcium channels. Activate the calcium channels, you now have calcium rushing into the cell, extending the depolarization, the length of depolarization. Now the longer the depolarization, the longer systole takes place. The longer systole takes place, the longer the contraction, the stronger the contraction. Now all of this right here is not as necessary, but it basically talks about how fast we can get calcium in and out. So any way you look at it, cyclic AMP, the main message of this, cyclic AMP, which is the intracellular messenger of epinephrine and norepinephrine on the muscle cells, will allow calcium into the cell faster, which again lengthens the length of depolarization, which strengthens the contraction.